So what I would like to do is commend you all on the challenge that we had of taking that online exam in such short notice. So that was something that was really challenging, and many of you were up for the task. The grades are not spectacular, but I saw a lot of positive stuff on there. It's one of those things that it's kind of a mixed bag. I purposely made the tests smaller because of the uniqueness of the situation. I didn't want it to impact your grades too greatly, so it was worth a lot fewer points. The downside of that then is that if you think of it from a percentage standpoint, you know, missing 5 points on a 50-point exam versus missing 5 points on a 100 or 85-point exam, it has more of an impact. So every point counts a little bit more, but every missed point counts a little bit more as well. But the essay questions are definitely getting better. There's still a lot of lack of detail, and that's one of the things that we're really going to hammer on this section. Today, our goal in lecture is practically solely to talk about the communication at the neuromuscular junction, which is our first step. And unfortunately, I can't get my iPad, I can't share my iPad the way I normally would. This is normally a lecture. It's one of my favorite lectures, but it is a very intimidating lecture because there's a tremendous amount of board work. So what I think I'm going to do, as I normally go over something twice, I think what I'm going to try to do this time is go over it 3 times. I think what we're going to do in this case is go through it with words first. I will describe it and describe it at the level of detail that I want us to have. But then I will also go through and we will do the visual of it. I'll do the drawing of it, and we'll talk about these processes as we go through it. Oh, and I can answer that question. That's a great question. And then we'll go through it with all the pretty pictures and all the pretty words from your textbook to hopefully make sure you get it. One of the important things to remember, one of the ways you're going to maximize your points on this class is to think about what is the immediate effect of something, right? What I'm looking for in this class is that telling me that you went to the store to get milk can be a completely correct statement. However, what I want is all the details. You put on your shoes and jacket. Then you grab the keys to your car. Then you unlocked your car, got in the car, started the car, drove the car to the supermarket, turned the car off, got out of the car, walked inside, grabbed the milk, went up to the cashier, paid with your credit card, went outside, got back into the car. That's what I want. That's what I want. I want what is the immediate effect, what is the immediate thing, the details. The details are how you're going to maximize your points. And while what I loved about the exam, the lecture exam, is that you guys are getting the big concepts, and that is the first step, understanding those big concepts. But what a lot of you are still lacking are those details, and so those are something that we're really going to try to hammer. The question that was asked before I started recording, there was a question about just clarification on the organization of the proteins in our sarcomere, in particular the relationship of the H zone and the M line and the A band. And so as I was explaining is that the A band includes everything where the myosin is. So we define the A band as being the part that contains myosin. Within that, some of that, the myosin and actin overlap. However, there are other parts where there is no actin. The actin does not overlap, and there's just myosin by itself. That is the part we define as the H zone. So the H zone is the part where the myosin and actin do not overlap, and that is contained within the A band. And then the M line is at the center, and it is where the myosin and the myomesin interact with each other. Again, it's still myosin, so it's still part of the A band. So the M line is contained within the H zone, and the H zone is contained within the A band. So we were just talking about that relationship. All right, next question. Will we be able to view our exams and see what we got wrong in our feedback? Yes. You should now be able to, if you go to the assignments and click on your exam, I believe I have it set up so that you can see your questions and your answers. So can I get a show of hands or a yes and a no as to whether or not you can see that? Here, let me clear that first. Give me yeses or nos, give me thumbs up, thumbs down, something if you can see your exams and have been able to check your exams and see your answers, because I believe I have it set up so that you're able to do that. A couple of you look like you've done it. Maybe a lot of you haven't done that yet. So I strongly encourage you to go and look at that. Again, I normally would not, especially with the lab exam, I normally don't allow the questions to come out, but this was such a unique situation. I want to see what you did right. I want you to see what you did wrong. So I am posting the questions. I am posting your answers so that you have a chance to look at that and figure that out, so that on the off chance we have to do it again, and I keep saying the off chance, but it is unfortunately looking more and more like we might have to deal with this again. I want to make sure everybody understands the process and what they need to do so that they can be successful. All right, so again, if anybody isn't able to see their exam or is having any problems with their grades or anything along those lines, please contact me. And what I'll do is I'll stick around a little bit after lecture in case anybody has any questions to answer those, or you can email me and we can either discuss it over email, or we can even meet privately in my Zoom room. In fact, I have my Zoom office set up now for office hours. It's the same phone number for my office number at work. So that's 916-484-8992. It's on the syllabus. So that is the Zoom room that I have set up as my default room. It's not the ones we're having the lectures in, but it is my default Zoom office, so that we can always meet in there if anybody has any questions or anything like that. All righty. Any other questions on any of that before we get started? All right, excellent. Then let me go ahead and clear those. Talk about our game plan for today. Here you see our game plan. Like I said, our lecture is going to be to start that big, long process of understanding how a muscle contracts. And like I said, we're going to divide it up into 3 easily digestible pieces. And our goal for today is primarily to focus on the first part of that, communication at the neuromuscular junction. So we need to talk about what the neuromuscular junction is, and then we can work on from there. We do have 3 assignments that are going to be working on during this section. The first 2 are your unit reviews. I've decided to, because again of the uncertainty of what our situation is, and to encourage you guys, this is actually a good point to talk about this. I talked with my 431 class yesterday, but the instructors and I in biology, all of us dealing with labs, have been talking a lot about how we need to get you guys to get the information to allow you to be successful in this class, because that is the real challenge. Obviously, as students, one of the things that you are immediately focused on is your grade, right? How do I get the grade that I want in this class? And certainly your grade is important. However, if your goal is to go on to nursing school or go on to somewhere else in the medical field, the other really important thing that you need from this is you need this information. Especially you guys here in the first semester class, the information that you're using right now, you're going to be using a lot in 431 when you get to 431. So if you're able to squeak by with an A because of the unique situation that we had here, but you haven't gotten any of this information, you are going to struggle mightily next semester when you get into 431. So it is really important that you learn this material. And one of those areas where it's going to be most challenging is in the lab component. I've done 3 now. While there are some quirks to it and those problems continue to expand, lecturing this way is not horrible. I don't get the interaction and the feedback that I normally enjoy in the classroom working with you guys. I don't get to work on the board, although I do have my iPad set up. I can't share it the same way I did before, but I'm going to try to use it to do some drawings today that aren't too hideous. But getting the opportunity to learn the material, especially the anatomy, the onus of your success is falling much more on you guys, right? The growing pains are over. I can't spoon feed you the information the way we normally would. So the onus is going to fall much more on you guys to make sure that you are successful in gaining the information. I will try to provide you with as many resources as possible and try to help you along the way. But your ultimate success in this class, and more importantly, your success moving forward, which is more important than your success in this class, is falling squarely on your shoulders. So this is a level of responsibility that we don't normally have to deal with, but you guys are going to need to work on to be successful. To encourage you guys to take up that mantle, I've decided that I am going to have you turn in the unit reviews on their normal due dates. So our Unit 10 review is going to be due, it looks like 1 week from today. And your Unit 11 review is going to be due online on April 2nd. To do this, what you can do, if you have the paper copy, you can, if you want to scan them and make them PDFs, that is fine. But I have the submission boxes set up now so that you can drop them in the drop boxes. And if you just want to take pictures of them and submit the pictures of them once they're completed, that is fine as well. So those are due on those days there. And then you have your PhysioX exercise that you're going to be doing, which again is a tremendous amount of fun. But especially because we don't get a chance to do this stuff in the classroom, you'll have the opportunity to work with that as well. And then I'm going to show you some of the tools to help you to be successful when we're working on the gross muscle anatomy, the origins, insertions, and actions that we're going to be doing. And again, I can walk you through it, not as easily as I can in the classroom. So like I said, it is going to fall much more on you to learn this material and retain this material, because this can be one of the more challenging sections. There's a tremendous amount of physiology and there's a tremendous amount of related anatomy. In some way, the bones were simple. It was bone, bone feature, bone, bone feature, bone, bone feature. For every muscle now, there's basically 4 things you need to know. Obviously, you need to know the name of the muscle, but for many of the muscles, you also need to know their origins. You need to know their insertions, so you need to know their attachment points, many of which are the bone features we just learned. And for all the muscles on your list, you need to know their actions. And using the correct term, just saying lifting the arm or rolling the shoulder or things like that isn't what's going to work. So we need to use that appropriate terminology, like flex, like abduct, like elevate, like protract. So we will work on some of that together, but like I said, you're going to be responsible for working on this on your own. So that's our game plan. We're going to start our lecture, start our discussion of the communication at the neuromuscular junction, and then we will learn about the tools that you can use to be successful as you're learning about the gross muscle anatomy, the origins, insertions, and actions. And like I said, we have 3 assignments. I have added those assignments to your module. So the same way the PhysioX has in the module of PhysioX has a Dropbox where you can drop the PDFs of that. You can also drop the pictures or PDFs of your unit reviews. Again, you can do that anytime from now till when they are due. All right, and again, this will be, I am recording the lecture, so you'll have this notes always at the top of it at the beginning of the lectures to remind you if you forget any of this. All righty, questions on any of that? Excellent. Oh, go ahead. Was there a question? No, just take up then, apparently. All righty, let's go ahead and get started then. Come on, there we go. As we talked about, there are going to be 3 main steps that we are going to break our muscle contraction process. Again, remember, it's a continuous process, but we are artificially breaking it up into 3 steps. And this would be a good place for me to remind you of those. Step 1, which we are going to be going over today, is communication at the neuromuscular junction. And one of the things that can help you with these processes when you're describing them or talking about them is to think in terms of what is the goal of each of these steps. The goal in this case is to convert a neural action potential into a muscle action potential. So that is step 1, and that is our goal of step 1. Step 2, of course, as we talked about, is excitation, contraction, coupling. Again, the excitation is the action potential, the big electrical signal. Contraction is what we want to do. We want to be able to generate that force. So we need to bring these two things together. It's a fancy term, but really all it means is our goal is to move our regulatory proteins. As we talked in the last lecture, there are three main types of proteins we find in our myofibril. They are our contractile proteins, myosin and actin. They are our structural proteins, like the Z disc, like the myomesin, like the titin, and our regulatory proteins, troponin and tropomyosin. And of course, as we talked about, the wonky thing that we need to do is to get this muscle to contract. And the way we move the regulatory proteins is by using calcium. So let's cheat and go back to the beginning here. And so we're going to use calcium to move the regulatory proteins. That leads us to step 3, which is the contractile cycle. And remember, we can also call it the sliding filament theory. So that's sliding the filament theory, again, where we have the myosin and the actin gliding over the surfaces of each other. It is a continuous process, which is why it is a cycle. And again, our goal here is going to be to generate our force. Well, actually, let's say it this way. Change the shape of the muscle cell. And that is going to generate our force. Or we can say it is produce our tension. And it is that tension, that force, that is going to move our body through space. So that's our goal. That is our game plan, to talk about and identify these 3 steps. As I mentioned, for all 3 of these steps, we are going to talk about it 3 ways. We are going to talk about how we start the process. And again, we want to make sure, again, I'm always trying to be precise with my vocabulary. So I will say initiate the process. But again, that's just a fancy way of saying we start the process. We also can terminate the process, or again, stop the process, and modify the process, right? In some way, disrupt or change the process in some way. So for each of these 3 steps, there are 3 ways we're going to talk about it. We can initiate it, which basically means turn it on, terminate it, which means turn it off, or modify it, where we disrupt or change the process. One of the ways we can really know we understand something is by messing it up. And so that's what we're going to do. We're going to talk about ways that these processes can get messed up. All righty, so that is the game plan. Again, we talked about that at the end of last class, and that's where we pick up here. Any questions on that? All righty then let us talk then. Go ahead and clear my drawing. And we can talk about the neuromuscular junction. The neuromuscular junction is basically our area of communication. Again, our goal is communication between our somatic motor neuron. Again, whoops, hold on, I want that. Remember, as we talked about, our skeletal muscle is controlled by our somatic motor nervous system. Luckily, they both speak the same language, and that language is an action potential. So when we make the decision to stimulate a muscle and get that muscle to contract, we start by generating an action potential in our, in our nervous system, and then we have to transfer that action potential to our muscle cell. Now, if this was just the wiring of your house, that would be easy. because all the wires in your house are hardwired together. So when you click a switch, that electron moves along the wire, goes all the way up to your ceiling fan and turns the light on, or turns the fan on, or it goes all the way to your garbage disposal and turns that on. We have those things hardwired together. But as it turns out, hardwiring things together isn't necessarily the most efficient way to do these things, right? As we've talked about many times, one of the scariest things in the world is, of course, a bear with an axe. If I stand here with my mouse in my hand and tell you I'm going to kill you, with social distancing and the fact that we're not anywhere near each other, you're not going to be too scared about that. However, if a bear with an axe busts in your door, looks you in the eye and says, I'm going to kill you, you're going to respond very differently to that. You're getting the exact same stimulus. You're hearing the words, I'm going to kill you, but you need to respond to them very differently. So with our nervous system, we don't necessarily want things hardwired together. We don't want the same response every time. We want to be able to make decisions. We want to be able to modify. And so typically, when a neuron communicates with its target cell, it does that at a structure we call a, a synapse. A synapse is a region of communication between a neuron and its target cell. So synapse is the general term. So again, this is a general term. Whereas the neuromuscular junction, and I will go ahead and emphasize that up here by putting a big circle around it, the neuromuscular junction is the specific synapse between our somatic motor neuron and our muscle cell. Let's take a look at that. Now, here is the pretty picture from your textbook, but I'm going to do a quick drawing for you first. So let's sneak back to our whiteboard. With our whiteboard, and I'll go ahead and leave that on there for now because it doesn't affect me. We need to do some quick basic anatomy of a neuron. We will get to the nervous system a little bit later in this class, but for right now, a neuron, like most cells, has a big centrally located cell body. This is where you will find the nucleus. This is where you'll find the rough endoplasmic reticulum, all the other stuff you expect to see inside of a cell. But our neuron has processes. In fact, it has two kinds. Processes should sound vaguely familiar because we just finished talking about processes in the skeletal system. What do you need me to say again? Can you be more specific?
Yeah, when you were going over the synapse, the region of communication between a neuron and a target cell, you said something about one is a specific synapse and synapse is just a general term.
Sure. Think of it this way. Bone is a general term, or joint, even better. I like joint better. Joint is a general term where two bones come together. All right. The term synapse is a general term, which means where a neuron communicates with its target cell. And that's actually what I'm about to draw right here on the whiteboard. So that is a general term. However, the knee is a specific joint. It is the specific joint between the femur and the tibia. The neuromuscular junction is a specific synapse. It is the specific synapse between our somatic motor neuron, which I'm about to draw, and our skeletal muscle cell. So a synapse is the general term for where a neuron communicates with its target cell. The neuromuscular junction is the specific synapse between our somatic motor neuron and our skeletal muscle cell. Does that make sense?
Yes.
Excellent. So we have our cell body. Most cells have cell bodies. That's an excellent thing. But like I said, it has two processes, two kinds of processes. As we learned in bones, processes are basically things that stick out. The first type are small, elaborately branched processes. So again, I'll draw a bunch of these. These small, elaborately branched processes are what are called dendrites. And they are what receive the information. However, the other thing that our neuron has, ooh, and let's go ahead and use a big line, cool, is it has a big, huge, long, single unbranched process. And this single unbranched process is called the axon. Oh, but I made my axon a little too long. Cheat. Oh, no. Okay, that didn't work. We'll try it again. There you go, because I need some room to play with. That axon is a single unbranched structure. Some of the axons in your body can be a meter long. You have cell bodies in your spinal cord, in the small of your back, whose axons go all the way down to your big toe, so that you can move your big toe, or so that if you step on a tack, you receive that information that you've stepped on a tack and you've hurt yourself. So these axons are are long, unbranched structures, and they are what send information. So dendrites receive information, and our axons send information. Their job is to send it to our target cell. Here is our target cell, and of course, in this case, that target cell is a muscle cell. Or let's be more specific, skeletal muscle cell. So it's long, cylinder shape. I'll cheat and put some stripes on it. I know they're supposed to be perpendicular, but again, I'm not going to take the time to do them right. You get the idea. Just pretend I'm drunk. There we go. We have that. Heck, we'll even throw a couple nuclei on there because it's multinucleated. Go. Perfect. So that is our skeletal muscle cell we need to communicate with. And what happens to our axon is, while it stays single and unbranched for the majority of its distance, when it finally gets to its destination, it is going to branch. And these branches are called axon terminals. And then these axon terminals end at a specialized structure I'll just all three of them. And these specialized structures are called synaptic end bulbs. So we haven't talked about the anatomy of a neuron yet, but these are the things we need to know. We need to know that it has a cell body, it has an axon, and most importantly, it has axon terminals and synaptic end bulbs. It is here. Now, as you look through your textbook, you will see different terms for these. These are sometimes called synaptic buttons. These are sometimes called synaptic boutons, terminal boutons, telodendria for the axon terminals. There are a couple different terms that will be used. But for consistency's sake, I will always use axon terminals for the branches of the axons, and synaptic end bulbs, which are the specialized, and let's cheat, give myself, these synaptic end bulbs are the specialized ends of our axon where it is going to communicate. So basically, what a synapse is this part right here. Let's do it that way. Where our neuron communicates with its target cell. This portion right here that I've got the box around is a synapse. Now, if this is a somatic motor neuron, and a skeletal muscle cell, then this specific synapse is our neuromuscular junction. All right. With me so far?
Yes.
Yeah. It only gets worse from here. So if this doesn't make sense, now is when you need to speak up, because we are adding more detail. All right, so I've drawn this picture. Notice I drew it sideways because it fit better on the screen that way. But notice if we sneak back here to our slide presentation, we see that part that I put the box around. Notice here is my synaptic end bulb. Here is the wall, the plasma membrane, the sarcolemma of our muscle cell. And notice there's a space in between. No, the somatic motor neuron is a special type of neuron. So the neuromuscular junction is a special type of synapse. So neuromuscular junction, like we see up here, is part of a special type. Where's my annotation? There it is a special type of synapse. All right. Now, as you can see, and as we're about to show here, the synapse has actually, whoa, did not mean to do that. So let's cheat and go back, because I want to write this out first. I'm going to write it out. I'm going to try to do as much writing as I can, like I would on the board, and then we'll go through it, the words. The synapse basically has three parts. All right. The first part, as we talked about, and let's say it this way, has three parts, and all three parts are specialized, have specializations. Part one is, as we just finished talking about, our synaptic end bulb. It is the end of the axon. So again, it's the, let's say it this way, end of the neuron. And it basically has three specializations that we see here in the illustration. The first specialization is it has lots of mitochondria. And those mitochondria there are, of course, there to produce ATP, because communicating with our muscle cell requires a tremendous amount of energy. Now, as I mentioned, the wires in your house are hardwired together. We have a big electrical signal that goes down the neuron. We have a big electrical signal we want to produce on a muscle cell. But as you see from the illustration, the two don't touch. There is a gap in between. And is that how you'd want to wire your house, putting a gap between all the wires so that your electrical signal could just arc or jump from one wire to another?
No.
No, that's probably not how you'd want to do that. So what we need to do instead is find a different way to communicate. And that different way that the neuron communicates is by actually using a chemical signal. So the second specialization that we see here in our synaptic end bulb, the second specialization are a large number of vesicles that contain a chemical signal. Now, that chemical signal, the general term, is a neurotransmitter. So the general term for that is a neurotransmitter. There have been over 100 different chemicals that have been identified that neurons can use to talk to other cells. However, here in the neuromuscular junction, there is only one and only one neurotransmitter that is used. And that specific neurotransmitter that is found here in our neuromuscular junction is a chemical called acetylcholine. As you can see, acetylcholine is a mouthful to say and to write out, but the good news is on your essays, once you've written out acetylcholine once, you get to abbreviate it ACH. This acetylcholine is how our neuron is going to tell the muscle cell to contract. It always has acetylcholine inside of it, but we don't always want to be releasing it. We want to control the release of it so that we only tell the muscle to contract when we want to. Well, telling the muscle to contract when we want to sounds like a wonky type of thing. So not surprisingly, the way we're going to tell the synaptic M bulb to release its acetylcholine is with the use of calcium. So the third specialization of our synaptic M bulb is voltage-gated calcium channels. Now, that gives us something we need to unpack. I think I can do this with a drawing. We'll cheat. Hold on, I want a solid one. And a line. All right, so this here is our channel, and it is a calcium channel. So guess what passes through a calcium channel?
Calcium.
Calcium, absolutely. However, this is a gated channel. What that means is that these gates can be opened or they can be closed. Now, because of the need of simplicity for this drawing, because I'm limited on what I'm able to do easily, I'm going to just use one gate to put one gate across the middle of this. But in reality, there are actually two. But this will help me to do, I think this will help me to do what I want to do. No, that won't work. Okay, well, I can cheat anyway. There is a gate in this, and that it allows this channel to be opened or closed. So it is a gated channel. And when you have a gated channel, you need to know what the key to unlock it is. In this case, the key that unlocks it is the voltage. It is a voltage-gated calcium channel. So calcium is what passes through the channel. The key to open it is a voltage change. Now, one of the things we talked about way back near the beginning of the class is that every cell has a resting membrane potential. Anyone remember what that resting membrane potential we said was? I know it's been a while. Was the inside of the cell positive or negative? Ah, perfect, excellent. It is negative 70 millivolts, excellent. The inside of the cell is very negative compared to the outside. So what happens is, with a voltage gate, if we can make the inside positive, then the gate opens. So if we can make the inside of it positive, then what's going to happen is that gate is going to, and again, I can't appear to be able to just swing one end of it up, although that would have been awesome. The gate is going to, can't figure out how to rotate this. No, well, doesn't matter. I'm not going to rotate it. But what'll happen is the gate swings open. Now, do you think any old positive change, just getting it to negative 69, do you think that's going to be enough? Probably
No.
not. Yeah, or 68 or 67. But there is going to be a number. There is a number where we can get it positive enough. And that point where a voltage-gated channel will open, that point we call the threshold. The threshold is the point where a voltage-gated channel will open. And typically, for most of these cells, that is negative 60 millivolts. So notice it's not a huge change. We just have to make it 10 millivolts more positive, and we can open that gate and allow the calcium to come in. All right. So that is the first component of our synapse, the synaptic M bulb, and it has three specializations. And again, I will highlight them here on the picture now that we've written them out. Here is our synaptic M bulb. Here are our vesicles containing our chemical signal. The general term for that chemical signal is a neurotransmitter. In this case, this particular case, this neurotransmitter is acetylcholine. Second specialization are large amounts of mitochondria, because releasing this chemical requires a lot of energy. So having mitochondria there to break down sugars to make ATP is going to be really important. And to control the release of that neurotransmitter, we have voltage-gated calcium channels. Again, these are calcium channels. We know there's more calcium outside the cell than inside the cell. So calcium wants to go inside. However, we want to control it. So the way we control that is with a voltage gate. If we can make the inside of the cell positive enough to reach threshold, to go from negative 70 to negative 60, that will open our gate and allow calcium to come in. All right, and then that calcium coming in is going to trigger the release of our neurotransmitter. All right, questions on that? Excellent. Let's talk about the second component of our neuromuscular junction, and that is our target cell, but not just any. If we look closely at our muscle cell, if we went back to the previous picture, I just drew that big long cylinder, but here we have a special part of our muscle cell, and that special part is called the motor endplate. So this is basically the connection point of the muscle cell. And that's this wavy thing that we see down here. In fact, that is one of its specializations. One of its specializations is it has a large surface area. If we want to allow a chemical to be able to communicate, we want a large surface area. So notice it has lots of undulations or invaginations in it that allow for that big surface area. Notice that gives us more surface area for the chemical to communicate with. But notice also, and I'll cheat and go back to my spotlight here, it gives it lots of nooks and crannies where that chemical can get trapped. And the longer the chemical stays here, the more likely it is going to be able to interact with the receptors. And that is the second specialization. Our second specialization are chemically gated cation channels. There. Excellent. Notice again, it is a cation channel. Being a cation channel, what is a cation again?
Positive ion.
It is a positive ion, absolutely. So this is a channel that allows positive ions to move into or out of the cell. There are a lot of positive ions, but what are the two most common ones that we've talked about?
Calcium.
True, but remember, calcium makes cells do wonky things. What are the other two positive ions that we've talked a lot about so far?
Sodium.
Potassium.
There you go. Sodium, which is a positive, and potassium, which is positive. Calcium is super important. But remember, as we've talked about, calcium makes cells do wonky things. So in this case, we don't necessarily want it to be calcium, but it is going to be sodium and potassium. So sodium and potassium are going to be the two ions that are going to move either into or out of the cell. And in this case, this is a chemically gated channel. So in this case, it's a channel that needs a chemical to unlock it. And guess what the chemical that is going to unlock this channel is called? Acetylcholine. Acetylcholine is what binds to it, is what opens. Oops. So it is the key. Now, one of the terms, again, your textbooks or sometimes the lab manuals will talk about is they will say ligand gated. Ligand gated just means chemical. Ligand is a chemical. So ligand gated, chemically gated means the exact same thing. Chemically gated makes more sense to me, but if you're a big chemistry buff and you like the term ligand, then you're welcome to use that as well. But the key here is this is a door that is going to be unlocked by acetylcholine. But remember, it's not acetylcholine that goes through it. It is sodium and potassium that goes through the door once the door is unlocked. All right. So those are the two specializations. Yep. So that's what the acetylcholine is. Will I use chemical on the exam? Yes, I will use chemically gated ion channel on the exam. Absolutely. The only reason I throw ligand out there is some of the textbooks still use that term. And so I'll throw it out there so it won't confuse people. But I will always use chemically gated. This will be the last time I use ligand when I'm talking to you about this. All right. So that is our motor end plate and its two specializations. Our third component of our synapse is, as you can see, the two don't touch. The synaptic end bulb and the motor end plate do not touch. So instead, there is the space in between, and that space in between is called the synaptic cleft. So this is basically the space between the two cells. And that is our third component of our synapse, of our neuromuscular junction. Not surprisingly, there are going to be some specializations in here as well. The first is obviously this is outside of the cells. It's outside of the neuron and it's outside of the muscle cell. So it is filled with interstitial fluid. Again, this is important because as we've talked about, the inside of cells has a lot of calcium in it. A lot of potassium in it, whereas the outside of cells has a lot of sodium. So our synaptic cleft would have a lot of sodium, low levels of potassium, different from the cell. But the other thing that is here in the synaptic cleft is, again, one of those alphabet soup terms. And that is acetylcholine esterase. Now, again, the good news is once you've written out that alphabet soup term once, you can abbreviate it ACHE with a big capital E there. And with the suffix of ACE, it should tell you this is an enzyme, and this is an enzyme that breaks down acetylcholine. Because after all, when we release that acetylcholine and tell that muscle to contract, we don't want it to contract forever. We eventually need to break down those keys so we can close those doors so that the muscle can relax again. So our neuromuscular junction, this synapse, has three main parts. The synaptic end bulb, that has mitochondria, has vesicles containing our neurotransmitter, and in this case, that neurotransmitter is acetylcholine. And our synaptic end bulb has voltage-gated calcium channels. Our motor end plate is the special connection point on the skeletal muscle cell. It has a large surface area with lots of invaginations on it, and it has chemically gated cation channels. Again, this is going to allow sodium and potassium to either enter or exit the cell, and it's chemically gated, so it is going to be opened by our acetylcholine. Right? Remember, our voltage-gated calcium channel is opened by a change in voltage. We reach threshold, and that opens to let the calcium in. And the third component is the synaptic cleft, the space between the two cells. Again, it is filled with interstitial fluid, and in that space, it has acetylcholine esterase, which is our enzyme that is going to break down acetylcholine. All right, so that is me writing it out. Let me go ahead. If everybody's got this, I'll give it one more second. And again, remember, I am recording this, so you'll all get a chance to look at this again. I'm going to go ahead and clear that so we can see the words that I have on the slides. All right, so any questions before I clear this? Excellent. So, nope, I can put it back. We got it?
I actually had a question. You said that the voltage gated calcium channel. That's old. That's the only one. That's what it's called, and that's the only one that's open by the change in voltage, right?
That is correct. Voltage gated channels, and again, this is not the last voltage gated channel we will run into in this class. We will see lots of others, but yes, voltage gated channels. Again, they're big alphabet soup type terms, but often the name tells you everything about it. Voltage gated just means that the channel can be open and closed.
Okay.
Voltage gated means that it is a change in the membrane potential that is going to open it. Chemically gated means gated, it is a channel that can be open and closed, but chemically gated means that it is a chemical that binds to it that causes it to open and close. Right? If you think about it, a voltage gated channel is kind of like those pressure things. Again, I don't even know if they still have these anymore. Back in ancient times when you went to the grocery stores, there were these pressure plates outside of the grocery stores. And you would step on that pressure plate, and that would basically, the pushing down of it would generate an electrical signal and would cause the door to open. Right? As a little kid, you used to jump on them all the time, right? Piss off your parents, piss off the grocer on the inside. But it was basically you were generating an electrical signal, and that electrical signal opened the door. Chemically gated is basically when you put the key in the hole and turn the key. You put that key in the hole and turn the key, and that is what causes it to open. Now, what causes it to reach threshold is exactly what we're going to talk about next when we talk about the process of that. But you have the right thing. That's the other thing too, right? If I just take a single can of soda and put it on that pressure plate, that isn't going to be enough to get it. And in fact, as a little kid, right, that 2-year-old goes over there and just walking on it typically isn't enough. So what does that little kid have to do? Jump up and down. And when they jump up and down, they produce a big enough signal. They reach threshold, right? If you don't have any idea what I'm talking about, then during the break, which we're about to take, what you can do is go into the bathroom. After you go to the bathroom, what I want you to do is just using your pinky, just put the littlest bit of pressure on the handle for the toilet. And just the tiniest bit of pressure isn't enough to make it go down. But maybe you put 2 fingers on there, put a little more pressure, 3 fingers on there. Eventually, you put more and more pressure on that handle, and eventually, boom, the handle turns down and the toilet flushes. There is some critical point you have to get to, and that's what threshold is. So yes, how we change the threshold, how we change the membrane potential in the neuron is going to be exactly how we describe the process of this communication at the neuromuscular junction. In fact, it's the second step. Yes, so I'm going to talk about all of this in much more detail. All right, but we just need to know the pieces first. Once we know the pieces, once we know the anatomy, then we can understand the physiology of how it functions. That's what we can talk about next. All right, so questions on these structures. All right, if we understand these structures, then, like I said, let's go ahead and clear my drawings, and we'll just do the same thing. Synaptic embulb, motor endplate, synaptic cleft, right? Those are the 3 components. We've already talked about the specializations of them. And like I said, we have a neurotransmitter that is used to communicate across the space. There are over 100 that have been found, but here at the neuromuscular junction is only acetylcholine. And the motor endplate has those chemically gated cation channels, which are basically what the acetylcholine binds to. And that is what allows us to have our communication. Your book does a really nice job of describing this process, and we'll go through all the pretty pictures in detail. And like I said, we're going to go through it 3 times. I'm going to do it first in text, writing it out, so that we can talk about that. And let me cheat and go back to that other picture, because I think that other picture looks nicely for it. Or maybe I'll switch, make a new slide that I can do that on. Then I'll actually do my own drawings for it. And then we'll go through the whole process with all the pretty pictures and all the pretty words from your textbook to make sure it makes sense. All right, but that is going to be a tremendous amount of work and effort. So what I think we need to do is take a break. Based on how things went last time, but also comments that I've gotten, emails that I've gotten, we will take a slightly longer break than just the 5 minutes we were taking before. I'm not going to take the full 15-minute breaks like we were taking in classroom, because we need to get through this information. But what I'll do is we'll split the difference and take a 10-minute break. So we will take a 10-minute break. Where's my phone? My phone says it is 12:57, so let's come back at 1:07, and at 1:07, we will pick up from there. And I will remind myself to start the recording again, because I'm going to pause the recording during the break so that we just don't have 10 minutes of dead air. So I will pause that recording, and then we will pick back up in 10 minutes. All right, any questions? All right, see you in 10 minutes. I'm recording. Before we dive into the material, a quick question that was asked during the break. If the face classes are not available and our exams will be online, will the curve our grades as was previously? I don't have a definitive answer to you for that. What I do know is that, again, I tried to de-emphasize this previous exam by decreasing the number of points that it was worth. And so as such, the curve becomes more challenging, because then it's worth even fewer points, and it becomes even less and less significant. So I probably will not be curving these scores. However, if we continue online moving forward, then obviously there will be some changes that are made to the grading criteria for the class, possibly emphasizing more maybe group discussions or participation type grades or things along those lines for being active in this online format, or something along those lines to offset the grades of the score, so that that the grades on your exams are definitely important for determining your grades, but there are other ways to emphasize and to maximize your points in this as well. Obviously, with more time, I can make a more vigorous exam, and with a more vigorous exam, then there's also the potential for them being worth more points, and then a curve seems warranted at that point if needed. So we'll see. But yes, obviously, as we move forward, the grading criteria is the more we stay online, the more the grading criteria is going to change. So there'll be ways that we can do that. I pretty much have been grading papers nonstop, because the class that my 431 class had their exams literally the day before campus got closed, and then you guys had your exams. So I really haven't had a chance to look at the long-term implications or modifications we would want to make if we stay in this format. Till literally last night. Last night I finally finished grading your guys' stuff, got it all input. So now I'm finally getting a chance to catch my breath and look forward to see what we're going to be doing from here. So yes, I don't have a definitive answers, but yes, the more we stay online, the more I will modify the grading criteria as it is appropriate. All right, any other questions? Raise your hands, of course. I should have asked this before. Make sure you can hear me. If you can hear me, raise your hands. So I want to make sure there aren't any problems before I get started. Excellent, excellent. Perfect. All right. All righty. Let's go ahead and get started then. So, as I mentioned, our goal now is to talk about communication at the neuromuscular junction. And as we mentioned, there's basically three ways we want to do this. First, we want to talk about initiation of communication at the neuromuscular junction. And absolutely on the exam, that is how I would ask the question. Describe the process of initiation of communication at the neuromuscular junction. Now, again, this wouldn't be a part of the question, but one of the important things for you to remember is to always think of in terms of what is the goal of this process. And again, I may use some abbreviations when I'm typing this all out to make it a little easier. But remember, as we talked about, basically our goal of initiation at the communication of the neuromuscular junction is to convert a neural action potential into a muscle action potential. That is going to be our goal. And as I mentioned, we will go through this process 3 times. We are going to do it once here I will talk about it and we'll write it out, looking at the pretty picture and talking about that. We'll go through it with all the pretty pictures and all the pretty words from your textbook. And depending on how time is going at the end, I may do the drawing of it that I've done in the classroom pretty much since the beginning of time. There are actually some pictures from last year's lecture actually on Canvas I have not actually released them yet because I didn't want you to be confused and or intimidated by those pictures, but I will post those as we go through, if we get a chance to talk about it and go through and do the drawing and discuss it that way as well. But what I want to do first is to talk about the process, write out the process as we look at the picture and make sure we understand it, and then we'll go through it again. As I mentioned, the way you're going to maximize your points on this particular exam is by making sure that you have the specificity, you have the details to it. So the important thing to think as you go step by step through this process is to ask yourself, what is the immediate effect of X? All right, so when something happens, what is the immediate effect of that? And what is the immediate effect of that? And what is the immediate effect of that? So when we communicate the neuromuscular junction, our goal is to, again, get a muscle to contract, more specifically, a skeletal muscle to contract. So, of course, the very first step in that process is the decision. We make the decision that we want to contract a muscle. All right, that is going to be the first step. Let me give myself a little bit more writing room there. Perfect. So for all of these, when we're talking about the very beginning, because it is skeletal muscle, because we voluntarily control it, it is always going to start with the decision. We decide we want to contract the muscle. When we make that decision, what happens is we generate a neural action potential. And again, I'll abbreviate that NAP as we move forward. And when we generate that neural action potential, let's do it this way, comma. The neural action potential travels down the axon. So again, it started, remember, on the contralateral side of my brain. Over here on the left side of the brain, I decided to remove my right finger. So I generate a neural action potential, and that neural action potential travels down the axon. And when it travels down the axon, it reaches my synaptic end bulb. Now, when we talked about the characteristics of, and so let's come in here with kind of a side note to 2. When we talked about the characteristics of muscle cells, we said muscle cells have elasticity, muscle cells have extensibility, muscle cells can contract. We also said muscle cells are irritable. They can receive a stimulus and respond to it. And we said they're electrically excitable. They respond by producing an electrical signal, and we called that electrical signal an action potential. At that time when we were talking about it, one of the things we mentioned, and I will remind you of that again, is that an action potential is a big positive signal. Oops. An action potential is a big positive signal. So basically, and again, one of the things we'll talk about even more when we get to the nervous system, you'll talk about it in physics, electricity moves a lot like water. So if I stand by the edge of my swimming pool and I take a big, huge, massive rock and I throw that big, huge, massive rock into the swimming pool, I produce a big, huge, massive wave. And that massive wave doesn't just stay there, it spreads down the pool. Well, that's basically what's happening here. I'm producing a big positive signal, that is the action potential, and it spreads down the axon. And when it spreads down the axon, that neural action potential reaches, and since I spelled it out before, I'll abbreviate it here, it reaches the synaptic end bulb. Now, if you're standing at the other end of the pool and I throw my rock in and produce a big wave, and that big wave reaches the other side, it's going to get you wet because I had a big, huge wave that I produced. And that's what happens here. I have a big positive signal that reaches my synaptic end bulb. And that big positive wave is going to make my synaptic end bulb more positive. So that neural action potential makes the synaptic end bulb more positive. Now, not surprisingly, we have a fancy term for this. The fancy term for this is that we say it depolarizes. And depolarizes is just a fancy way of saying that it makes it more positive. Now, this is a big, huge positive wave. Our synaptic end bulb, our neural action potential is 100 millivolts, big positive signal. So, not surprisingly, if we depolarize this, what happens is our synaptic end bulb reaches threshold, right? And again, in parentheses, its membrane potential goes from -70 millivolts to -60 millivolts. And of course, what is the immediate effect of reaching threshold? Why is that threshold number so magical?
It opens the calcium gate.
Absolutely, absolutely. It is going to, so that depolarization to threshold, oops, no, wait, where am I writing that? Absolutely. The depolarization to threshold opens, and I'm going to abbreviate it here for space, VG for voltage gated, CA for calcium channel. And actually, you know what? I lied. I'm going to write it out just because we're doing this for the first time. So the immediate effect is that it opens our voltage-gated calcium channels. And of course, what's the immediate effect of opening calcium channels? If you open a door for calcium, what's calcium going to do?
Make it do wonky things.
True.
How's it? An A's ending is a.
Yeah, it's going to enter the cell. Remember, there's a lot more calcium outside the cell than inside the cell.
Esterase, ASE,
Yeah.
that ending is a enzyme.
Why is it?
So, absolutely right. You guys are absolutely right. Calcium, the immediate effect of opening those voltage-gated calcium channels is calcium is going to enter the synaptic end bulb. Oops. Now, there is one more thing, and I'm going to cheat and draw it right here, that we didn't talk about that this cell has. One of the things that this cell has that we haven't talked about yet are calcium pumps. Not surprisingly, since calcium makes cells do wonky things, cells don't want calcium inside of them. So what happens is we have, and I'll cheat and draw an arrow here. It has this calcium pump. This calcium pump is going to use ATP. And it's going to use that ATP to kick calcium out of the cell. There we go. So it's going to kick calcium out. Right? We don't want calcium inside of the cell. However, that's one of the reasons why calcium levels are so low. However, if I've opened a bunch of these voltage-gated calcium channels, massive amount of calcium is coming in, right? If you're on a boat and on the boat, suddenly I drill 17 holes in the bottom of your boat and you have a little hand pump, you can pump some of the water out, but that water is going to come in a lot faster than you can pump it out. And that's what happens here. Our calcium enters into the synaptic end bulb. And so we get an increase, even though there are pumps. Whoops, no, hold on, let's go back to black. The concentration of calcium increases in the synaptic end-bulb. Right, even though there are pumps trying to stop it, the concentration of it increases in the synaptic end-bulb, and so we have more calcium inside. And as we talked about, calcium makes cells do wonky things. In this case, the wonky thing calcium is going to do is it triggers the release of acetylcholine via exocytosis. Now, exocytosis is the process by which these vesicles come to the plasma membrane and are expressing their contents. So it triggers that release via exocytosis. Oops. Now, again, the key word there is triggers. Calcium doesn't actually grab the vesicle and bring the vesicle to the plasma membrane and shove it out. There are actually 25 different proteins involved in that process. It is a huge, elaborate process. We could spend a whole lecture talking about, and I won't make you do that. But the key is it coming in basically knocks over that first domino, giving us this big, huge domino effect, which causes the vesicle to find the plasma membrane, attach to the plasma membrane, and release its contents. So it triggers the release of acetylcholine via exocytosis, and we should have actually finished that off there, where it's exocytosine 2 into the synaptic cleft. All right. Questions on that? Excellent. So now we have acetylcholine in our synaptic cleft. Now, again, remember, there is acetylcholine esterase, that enzyme that is going to break up the acetylcholine in the synaptic cleft. But again, we are releasing more acetylcholine into the synaptic cleft than that poor little enzyme can break down. So we get an increase in the amount of acetylcholine in the synaptic cleft. And of course, that acetylcholine isn't going to sit there and huddle against the wall of the synaptic end bulb, right, wanting its mommy. As we know, when there's a high level of that amount of stuff, then what is going to happen is our acetylcholine is going to diffuse across the synaptic cleft to the motor end plate. That's what we see here. Can you guys see my mouse when I do that? Yes, no, maybe? Yes, excellent, okay, perfect. So as you can see here, we have that acetylcholine coming out and it's diffusing across the synaptic cleft. And notice when it diffuses across the synaptic cleft, our acetylcholine binds to our chemically gated cation channel and opens. Now, when we open our chemically gated cation channel, cations are going to flow. However, as we talked about, there are two primary ones, sodium and potassium. Now, from what we know, which way is sodium going to want to flow? Does it want to go into the cell or does it want to go out of the cell? Into, excellent. There's more sodium outside the cell, so it wants to flow into the cell. And which way does potassium want to flow?
Out.
Out of the cell, excellent. There is more potassium inside the cell than there is outside, so it wants to flow out. Now, if you think about it, if one sodium enters the cell and it's a positive ion, and one potassium leaves the cell and it's a positive ion, one positive thing came in, one positive thing left, are we going to change the membrane potential of our muscle cell if that occurs? No. One positive thing going in, one positive thing coming out isn't going to change it. Now, while this cation channel allows both sodium and potassium to flow, there's one big difference. Well, two, I guess. One is the direction. Sodium comes in, potassium goes out. The other big difference is in this channel, there is going to be lots of sodium that enters into the cell and only a little bit of potassium that is going to flow out. So if you think about it, the net effect of this is that you have a lot of positive things coming into the cell, very few positive things leaving the cell. The net effect is our muscle cell gets more positive. And did we have a term that we used for when a cell gets more positive?
Action potential?
Well, we're going to get to the action potential, but if we think back up, like, oh, I don't know, for instance, in step 3, was there a term we used for something when it gets more positive?
Polarization.
Depolarizes, absolutely. So basically what happens is our muscle cell depolarizes. However, you were exactly correct before. And here is the key word, if. The key word here absolutely is if. It means it doesn't always necessarily happen. But if the muscle cell depolarizes enough, and how would we know if it depolarized enough? Would there be some critical point we would need to reach for something magical to happen? Didn't our synaptic bulb have something like that? If it depolarizes to threshold, bingo. If the muscle cell depolarizes to threshold, then the muscle cell produces a muscle action potential. It produces its own big positive wave. And just like the neural big positive wave, is that big positive wave going to just stay right there at the motor end plate? No. So it produces a muscle action potential, a big positive wave, at the motor end plate. And the muscle action potential then spreads down the sarcolemma. And there you go. Just that easily, in 14 simple steps, we have reached our goal of converting our neural action potential into a muscle action potential. Now here's the good news. This is the longest process. Not all of them are going to be 14 steps long. But this is the point that I want. This is what we need. Notice step by immediate step, we have everything from the point of where we made the decision that we wanted to contract the muscle to the point where if that depolarization is big enough to reach threshold, we generated our muscle action potential at the motor end plate, and it started spreading down the sarcolemma. So that is initiation of communication at the neuromuscular junction. All right, questions on that? All right, I got to delete this. I got to remove this. So, because again, I just used the annotation to do this. So I have to remove this when we move to the next slide. It's going to go away. So now that we've gone through this process once here, I'm going to jump ahead. So again, last chance before I clear it. Any questions? Again, remember, I'm recording this lecture. It is recording, right? Please tell me it's recording. Yeah, okay, excellent. So you'll have a chance to go back and see this again. So I'm going to go ahead and clear this. So we can look at the pretty pictures from your textbook that do the same thing. So we're going to jump past the next two steps. We'll come back to those and go through it again with all the pretty pictures and all the pretty words from your textbook. So again, initiation of the communication at the neuromuscular junction starts with the decision. We decide we want to contract the muscle, we produce a neural action potential. That neural action potential is a big positive signal that makes the cell more positive. And again, the fancy word for that is that it depolarizes or causes a depolarization. Yep, so this starts at the axon and this spreads down the axon and reaches the axon terminal, where it then reaches our synaptic end bulb. So yes, I'm gonna take a quick drink. That big positive signal starts up basically right next to the cell. It spreads down the axon, down here, the axon terminal, and finally to the synaptic end bulb. That big positive signal depolarizes the cell to threshold, and when we reach the threshold, our voltage-gated calcium channels open. And the immediate effect of opening those channels is that calcium rushes in. Again, there are pumps that are kicking it out, but those pumps don't do any good because more calcium is coming in than can be kicked out. That calcium coming in then triggers the exocytosis, causes these vesicles, again, 25 proteins involved, an incredibly elaborate process of having this vesicle come to the plasma membrane and release its acetylcholine by exocytosis. Once it's in the synaptic cleft, the acetylcholine diffuses across the synaptic cleft to our motor endplate. And here we have the cool picture from your textbook. Oh, I lied. There it says ligand gated because, again, your textbook uses that term. But here we see that chemically gated cation channel. Our acetylcholine is the key that is unlocking it. It is binding to the channel, but it is sodium and potassium that travel through it. Sodium that comes in and potassium that goes out. But as we talked about, there are big differences in how much. I think the technical term is lots and lots of sodium enter into the cell, but only a little bit of potassium leaves. So the net effect of that is that we depolarize the cell. And when we depolarize the cell gets more positive. And if it gets big enough, and of course the way we know it gets big enough is that it reaches that magical point threshold. If we can make the muscle cell positive enough to reach threshold, then it will generate its own muscle action potential. All righty. Questions on that? All right, I have one more question. If the signal is big enough, we get a muscle action potential, and that muscle action potential spreads down the axon. Is the end of communication at the neuromuscular junction. So it's the last step in part 1. But guess what generating that muscle action potential and having it spread down the sarcolemma is also? It's the first step in part 2. Remember, this is a continuous process. So where this, exactly where this stop part ends is where the next step is going to begin. All right. Questions on that? Again, if this doesn't make sense, you need to ask now because it is only going to get worse. Excellent. Blank stares mean we got it. So what we can do then is come back here and cheat and go back to my blank slide. Now that we are going to, again, and we have to continue the rest of the process of how we get the muscle to contract. But before we go on to excitation, contraction, coupling, before we go on to the contractile cycle, now that we've turned this process on, we know it is going to lead to a contraction. We don't know all the steps yet, but we know eventually the muscle is going to contract. The problem is we don't want that muscle to contract forever. We have to have a way to turn it off. And so that's what we're going to do now. Now that we've talked about how we initiate communication at the neuromuscular junction, we are going to identify how we will terminate that communication at the neuromuscular junction. Again, one of the easiest ways to think of this in terms of the goal. In this case, our goal is to get the muscle to stop to contract, obviously. That's the ultimate goal. But here, if you think about it, our goal was to produce a muscle action potential. So our goal of termination is to stop the muscle action potential. So now that we have started the process, we want to terminate the process. All right. So let's talk about the steps involved in that. Obviously, when we're talking about the movement of skeletal muscle, we need to start with the decision. At this point, we decide we want to relax the muscle. So that is going to be the first step in this process. Same way the first step in the process was deciding to contract the muscle. Here, our decision is going to be to want to relax the muscle. All right. Now, when we decided to contract the muscle, we produced neural action potentials. So what we are going to do here now that we want the muscle to relax, we are going to stop producing neural action potentials. Oops. So no new neural action potentials are going to be produced. That means no more big positive signals. Oops. Traveling down the axon to the synaptic end bulb. And what that means is that now the cell can't stay depolarized. Oops, depolarized. Cells want to be at rest. Its resting membrane potential of -70 millivolts. And not surprisingly, we have a fancy name for that as well. When a cell goes back to its resting state, we say the cell repolarizes. Now, if the cell repolarizes, and again, we want to think of what the immediate effect of this is going to be. We repolarize, we go back past threshold on the way down, and as we pass threshold on the way down, the voltage gated calcium channels are going to close. And big capital letters here. No, oops, new calcium into the synaptic end bulb. Oops. So no new calcium coming in. That helps, but it doesn't actually end the process. Because if you think about it, there is already a bunch of calcium in the cell, in the synaptic end bulb. So what we need to do is we need to get rid of it. Luckily, we have those pumps, right? I can go ahead and, well, I won't bother drawing it, but you remember there was that pump that was here pumping the calcium out. Before, it wasn't having much of an effect because for every calcium it pumps out, 10 more were coming in. But now that the voltage-gated channels are closed and no new calcium is coming into the synaptic end bulb, now finally the calcium pumps can remove the old calcium from the synaptic end bulb. So all the calcium that was still in there, no new calcium coming in, and we get rid of the old calcium. And when that happens, we stop the endocytosis of acetylcholine. And so again, big capital letters, no new acetylcholine into the synaptic cleft. Right? Kind of what we saw with calcium. No new calcium coming in, but that wasn't enough to stop the process. We had to get rid of the old calcium. Well, here we have the same issue on our synaptic cleft. We have no new acetylcholine being released into the synaptic cleft, but the problem we have is we still, I'm not going to say still, we need to remove the acetylcholine, or let's say old, I liked old acetylcholine from the synaptic cleft. And as it turns out, there are 3 ways we can do that. The first, and the simplest if you look at this, notice if you look at it, these things are very close together. They're the nooks and crannies in the motor end plate to help to encourage the acetylcholine to stay here. But there is nothing stopping the acetylcholine from just sneaking out of the synaptic cleft and moving away. So one of the ways we get rid of it is just simple diffusion. The acetylcholine diffuses out of the synaptic cleft. That's one way we can get rid of it. The second way we can get rid of it is that acetylcholine is an important resource and our neuron needs a lot of it. So one of the things that will happen is our neuron can actually bind some of this acetylcholine and bring it back into the cell. Of course, exocytosis is how you express stuff. So binding something and bringing it in is endocytosis. However, as I'm sure you have guessed, the primary way we're going to get rid of that acetylcholine is that enzyme Acetylcholine Esterase. Again, once you write it out once, you can abbreviate it ACH, capital E. And its job is to break down the ACH. So whether the synaptic end bulb reabsorbs it, whether it diffuses out of the synaptic cleft, or whether our little Pac-Man gobbles up that acetylcholine esterase, the end result is there is no more acetylcholine in the synaptic cleft. Oops. Here things get a little easier. No more acetylcholine in our synaptic cleft. Then, of course, our chemically gated cation channels close. With them closed, there's no new, no new sodium entering into the cell. Remember also, there's no new potassium leaving either. But remember, it was the large amount of sodium coming in that caused the cell to depolarize. So the sodium has a much bigger effect than the potassium does. With no new sodium entering into the cell, our muscle cell that's not an else. goes back to rest, or again, we can use the fancy term, repolarizes. And if our, oh, I did the same thing there. Our muscle cell repolarizes, and if our muscle cell repolarizes, it stops producing muscle action potentials at the motor end plate. Oops. And boom. At the motor end plate. And as a result of that, we have reached our goal. We have stopped the muscle action potential. And I wrote over my picture a little bit there, but hopefully you guys can still read that enough to make sense of that. Or maybe if I grab it and move a little bit, that'll help a little bit. There, hopefully it's not too bad there. I can't appear to figure out how to get back into a text to, yeah, so I can just put a new text over it. I can't fix that. All right, so there you go. We have now terminated communication at the neuromuscular junction. Our second possible essay question. Questions on that? Excellent. Then if this makes sense, I'm again give you one last chance to look at it, and I'm going to erase it. Going once, going twice. All right, clear that. Then let's look at the pretty pictures from your textbook on this. Again, I shorthanded it here because, again, normally in classroom we write it out on the board and we draw it out on the board. So again, here, this here on the slides isn't every single minutia of step. It's really highlighting the important concepts. And so as we've talked about, the first thing we have to do to get back to that initial state, to stop the muscle action potential. Hold on. Yes, all of this information is recorded and will be put on YouTube. So you will have the chance to look at this many, many times. And I actually have another resource that you should look at as well. So let's do this. Return to the initial state, remove the acetylcholine. Again, that's going to be our first goal. If we want to get the acetylcholine out of the synaptic cleft, the first thing we have to do is stop releasing it. We do that by stopping the neural action potential, repolarize the synaptic end bulb, and we close the voltage-gated calcium channels. We kick out the calcium, and without calcium, no new acetylcholine is released. But remember, as we talked about, we have to get rid of the acetylcholine that is there. One way is by diffusing out. We can use our enzyme, Acetylcholine Esterase, to break it down. And some of it will be endocytosed. Here we see the enzyme breaking it down. And again, without that acetylcholine, our cation channel closes. No more sodium coming in. No more depolarization. No more muscle action potential. All right. Questions on that? All right, excellent. Then here is the last thing that I want to do for today. Let's cheat and go back to here, and let's talk about modification. And let's do this too. Hold on a second. Let me. Hold on, because I want to change something. I want to escape there. Oh. I'm still here, hold on. I just need to fix something first. I want to cheat and make the picture bigger so we have more room to play with. All right, and then if we go back here and we, perfect. All right, can you guys, oh, hold on, I can't see you now. There's that. There's my chat. Perfect. All right. Excellent. All right, can you guys see that okay? Nice big picture. All right, the last thing we want to do is, if we really want to understand this process, one of the ways to really show that we understand the process is by messing that process up. One of the things that helps us with this is often what happens is there are toxins or poisons or other chemicals that have some effect. But as we understand how these things work, we understand we can use that to our advantage to be successful. Right? How many people here have ever heard of the bacteria botulinum before? You can raise your hand on the participation thing or give me a thumbs up or something like that if you've heard of botulinum before. A couple of you have, a couple of you have not. Botulinum is a bacteria often associated with foods that have been spoiled, inappropriately canned or jarred foods that have spoiled or become toxin that way. And botulinum produces a toxin called botulinum toxin. What botulinum toxin actually does is it's actually a very, very simple toxin. And what it basically does, and I guess I can cheat and do this here with it. Actually, let's draw it. Remember how I said in here the process of exocytosis requires 25 different proteins to get this vesicle to bind to the plasma membrane. And all botulinum toxin does is it breaks one of those 25 proteins. It is a protein actually called SNAP, S-N-A-P. You don't need to know what SNAP stands for. You don't even have to memorize the name SNAP. But what you do have to know is that all it does is breaks one protein. All right? Now, when we break that protein, let's think about what happens. We make the decision that we want to contract the muscle. We generate the muscle action, the neural action potential. The neural action potential comes down here to our synaptic end bulb, depolarizes our synaptic end bulb, opens our voltage-gated calcium channels, calcium rushes inside, and calcium tries to tell this vesicle to bind and release its acetylcholine. But because we've broken that one protein, our vesicle cannot bind to the plasma membrane and release its acetylcholine. So I'm telling my muscle to contract, but my synaptic end bulb cannot release acetylcholine. With no acetylcholine, I can't open my cation channels. If I can't open my cation channels, the muscle cannot contract. Basically, what we get as a result of that is what we would call a flaccid paralysis, where even though I'm telling the muscle to contract, the muscle cannot contract. Sounds like pretty scary stuff, right? And botulinum is pretty scary stuff. However, if you give it, a cutesy name like, for instance, Botox, then people have no problem at all injecting it into all parts of their body, right? You have those pesky wrinkles on your forehead, no problem. Just inject a little botulinum toxin into your forehead. It'll break those proteins, causing the muscles of your forehead to relax, right? Giving them that flaccid paralysis. Sure, you can't emote anymore, show people that you're surprised or happy or sad, but as long as your forehead is nice and smooth, that's all that matters, because as we know, it's only the outside that counts, right? That botulinum toxin is what we use and cosmetically as Botox. And again, there are valid uses of Botox. Botox is used as a muscle relaxant in the case of like smoothing wrinkles and things along those lines. But there are also certain types of migraines that can be caused by constriction of the smooth muscle in the blood vessels. And so what they found, these typically of the temporal region. And so what sometimes they will do is they will inject this Botox into your temporal region, paralyzing that smooth muscle. That smooth muscle dilates, more blood comes to the area of your cortex there, your temple there, and it can eliminate or dramatically minimize, migraines that way. Botox is used for paralyzed in other things as well. If you've got an eye turn for kids, they'll sometimes put Botox in there to weaken the muscle so that the eye comes back. Again, once we understand how these things work, we can use them for positive things. Now, is the effect last forever? No. We are breaking a protein though. So what has to happen is that neuron has to realize that protein is broken. It has to send a signal way back to the cell body to tell the cell body, hey, make more of that protein. We then have to ship that protein all the way back to the synaptic end bulb so that we can fix it. And that takes a lot of time. And that's one of the problems with Botox. You go to that Botox party and you get it injected into your forehead or your crow's feet by the side of your eyes so that your face is all smooth. But, you know, you're also drinking a bottle of wine while you do it. And maybe the needle gets injected a little too far and it ends up in your blood supply. And then once it's in your blood supply, it could go to everywhere, like, oh, I don't know, your diaphragm. And if you cannot contract your diaphragm anymore, what are you going to have problems doing?
Breathing.
Breathing.
Yeah, breathing, absolutely. But like I said, right, your neurons are located in your spinal cord. It's only 2 feet from your spine. Well, it's less than a foot. It's a foot from your spinal cord to the diaphragm, right? And it only takes those neurons. They can move a protein, as fast as a centimeter down an axon a day to get there. So how long is it going to take that protein to get to your diaphragm so we can fix it and it can start contracting again? Right? And how long can you hold your breath for? Right, not nearly enough. So again, that Botox can be very, very deadly, but now we understand how it works. What happens is it breaks a single protein, stops the acetylcholine from being released, and as a result of that, we get that flaccid paralysis. Another type of flaccid paralysis happens from a chemical known as curare. I think I'm spelling that right, curare. Curare is a toxin that is formed in certain plants. There are some frogs that secrete it. There's even a couple mushrooms that produce a chemical very similar to curare. Again, any good adventure film worth its salt, at one point or another, our protagonist is running through the jungle because they've stolen some golden idol from some villagers that he's going to be able to take back to his museum and get fame and fortune from. And of course, those villagers are chasing after him with their blow darts, right? And they're hitting all of his assistants and they're collapsing to the ground as a result of that. But of course, he dodges them all and lives happily ever after. Well, that toxin used in those types of blow darts is another type of toxin that causes another flaccid paralysis, and that is curare. What's interesting is curare works in a different way. Curare is a chemical that gets into the interstitial fluid. And I'm going to cheat here. Again, go to my drawing tool, get a couple circles here. What curare does is curare will come in and it binds to our cation channel. And the key with curare is that it binds and blocks. So again, if we think of this location as where we put the key in to unlock that door, if I take gum and shove that gum into the lock of the door, what's happening to that door?
Locked.
Yeah, it's not going to be opened anymore. And that's what happens here. So again, if you think of the same type of thing, we send the neural action potential, we depolarize, we allow the calcium in. In this case, we even release the acetylcholine. You could dump a whole bucket of acetylcholine here. But if that acetylcholine can't bind to the receptors, it can't stimulate the muscle, and the muscle can't contract, and we have a flaccid paralysis. Again, scary stuff, especially if it's affecting muscles like your diaphragm and things along those lines. But notice this one doesn't actually break anything. Remember, botulinum toxin breaks a protein. This one just gums things up. It's not actually damaging the neuron. It's not actually damaging the muscle cell. And so what they found is this is something we can take advantage of. In this case, what they do is, if you look at some of these incredibly complicated transplant surgeries, right, where people are reattaching hands, or some people have actually had face transplants, where they get a face from somebody else and they get it put on. Now, Your muscles, like right now while you're sitting in your chair, your leg muscles, you would say, are relaxed. However, if someone in your household was a paraplegic and you went and poked their legs, would their relaxed legs feel the same as your relaxed legs?
No.
No. Your legs are indeed relaxed, but there is a little bit of muscle tone in them. It helps to support the joints. It helps to hold things in place. And the reason that occurs is that your synaptic end bulbs will spontaneously release tiny amounts of acetylcholine, just enough to produce a tiny bit of tension in that muscle. Usually you don't see those twitches of the body, but it's just enough to provide that tone. However, if you're doing the delicate surgery where you're trying to connect blood vessels and connect nerves to someone's delicate skin of the face that you're trying to put on somebody else, even small changes in tension can cause major problems. So what they'll do is they'll take curare and they'll bathe the area that they're working with in curare. That paralyzes the muscle in that small area. It allows them to be able to do the work that they need to do. And then when they're done, they can just take their saline solution and rinse it out, and the curare goes away and everything functions normally again. So for some of these delicate transplant surgeries, they're actually using toxins like curare to their benefit. Now, you may or may not have heard of curare, you may or may not have heard of Botox, but most of you have probably heard of the last one, and that is tetanus. What is tetanus associated with? What can you get tetanus from?
Or sheet metal.
Yeah, there you go. Rusty metal or things, a rusty nail, things along those lines. Rust, absolutely. Tetanus produces a toxin that has a similar but very different effect to curare. Tetanus also will come in and it binds to my chemically gated cation channel. However, the big difference between tetanus and curare is tetanus binds and activates. Now, curare gums it up. Tetanus binds and opens it up. But like curare, tetanus doesn't like to let go. That means this channel opens with that tetanus toxin, and sodium is constantly coming into this muscle cell. And if sodium is constantly coming into this muscle cell is constantly depolarized, it's constantly producing action potentials, and it is constantly contracting. It produces what we call a rigid paralysis, where basically the muscle locks up. In fact, that's what tetanus is commonly referred to. Tetanus is commonly referred to as lockjaw. If you look back in some of the old medical journals, you see these amazing pictures of people whose bodies are just completely contorted into these incredibly painful positions because they have these massive, massive contractions of the muscles of the body that do not stop. They cause considerable pain, incredible agony. Some of the muscle contractions can be power enough where they actually break their own bones. And again, think of the effect on our respiratory muscles. With something like Botox or curare, where you have a flaccid paralysis, we can put you on a ventilator these days, and we can basically elevate and depress your rib cage so that you can breathe relatively normally. However, if all the muscles of your chest are all locked up rigidly, are we going to have an easy time ventilating someone like that? No, typically not. So it's typically going to be a very challenging type of condition to have that. So notice, by understanding how this communication at the neuromuscular junction works, we can understand how these toxins get in there and interfere or modify the normal function. All right. Questions on that? All right. I was thinking about this. I don't think, again, drawing it is totally for me. It helps me. I'm not sure necessarily if it would help you. However, I do want to remind you, you have some great resources that can help you to understand this information. So any questions on the modification before we move forward? Does Parkinson's disease affect any part of step 14? Oh, great question. So yes, one of the interesting things about Parkinson's disease is it is a disease of the nervous system. So it doesn't affect step 14 so much as it affects the release of acetylcholine. So acetylcholine release is more, what's the right term? Is less consistent. So you're absolutely right. One of the things that happens with someone who has Parkinson's is they tend to have resting tremors. So again, so that normal process of maintaining tone and posture is disrupted because it's less consistent in the release of acetylcholine. So you get more of a resting tremor. And also with some Parkinson's patients, when they do a controlled behavior, right, that tremor can go away. But sometimes also they can have those controlled movements can be intermittent, where they can start and stop as they're reaching out to grab something as well. So it isn't so much step 14 that interferes with it. It's really more affects the release of acetylcholine because of the problems with the neurons. Excellent question. Any others? All right, so I'm going to switch away from the slides because I want to take you to, where do I want to take you? I want to take you here. I want to take you to, yes. So yes, you absolutely need to know these for the exam. So if you think about it, what we did today, remember as we talked about, there are 9 possible essay questions. And I'll tell you right now, if there's not 9, it actually is closer to 7. There's 7 essay questions, 2 of which I guarantee will be on your exam. And these are the first 3. So on the exam, 3 of the possible essay questions are going to be describe the process of initiation of communication at the neuromuscular junction, describe the process of terminating communication at the neuromuscular junction, and then I could ask you some question one of 2 ways. What is it that tetanus does to communication at the neuromuscular junction? Or I could say describe 2 ways or 3 ways that you can modify communication at the neuromuscular junction. And you would talk about these toxins. All right. Now, where I want to go, let me get to the right place. Did I close it already? Okay, here it is. Got it. I found it now. Hold on. So now what I want to do is share that. Hopefully, you are taking advantage of the resources that are online for you, your modified master in A&P. There are some tremendous resources, as I said, as I talked about at the beginning of class, and we'll continue to discuss as we move forward. There is a huge need for you to take the onus, for you to take the lead in studying this, finding as many ways to make sure you master this material as possible. And this is a great resource. Here, there are amazing resources, art labeling, practice tests and quizzes, all of these types of things that can help you to be successful. However, one of the easiest ways to find the things that you are interested in is by going to the study by textbook chapter. So when you're in your study area of the Master A&P, can you guys see this? Again, I started this, before I go any further, can you guys see what I'm doing? Okay, excellent, perfect. Study by chapter, and then you can find the chapter you're looking about, and we're on muscles and muscle tissue. So that's the one I want. Notice there are chapter quizzes and practice tests. Hopefully this is not new for you. You guys have seen these things. We have the e-tests portions where it talks about the different things, and it even kind of helps you. We talked about the 3 types of muscle tissues and things along those lines. So we've done all of these. But what's really, really cool, and what I love for what we're doing right now, are these A&P flicks. These A&P flicks are great animations that go ahead and show this process. Now, I'm going to actually pause the recording during this because it's silly for me to record a video that you can watch online. So I'm going to pause this and we're going to watch this video and you're going to see how it does a great job. So rather than me drawing this, let's look at a nice example of this. So I'm going to go ahead and pause the recording before I forget. So that is one of the many great tools that you have here to help you to understand this stuff as you go through it. There's a great interactive physiology, which is a wonderful tutorial structure that goes through that talks about these things. So we have it for, again, interactive physiology for the events of the neuromuscular junction. It is a great resource where it walks you through it step by step. There's for metabolism, for all sorts of things. So there's great resources here. More video tutors for some of these processes, flashcard, glossaries, great resources. And this is one of the places where you can find your PhysioX that you're going to be responsible for as well. Notice here the link is to the 9.1, whereas if you go back to the home, and under the lab tools, that is where you can get to the PhysioX. That is the 10.0, which is the web version, and the HTML version, which is much easier to use. The other thing that you'll find under here on the lab tools, and I think we talked about this for the bones, but I want to emphasize it again for the muscles, is to come here to the practice anatomy lab. Aside from the fact that you have a flashcard maker that you can use, that will be very helpful for this as well. When you go to the practice anatomy lab here, there are two things that are really useful. The first thing that's really useful when we look at the muscular system, and we want to look at models, not at the cadaver, although the cadaver is fun too. But when we look at the models, for instance, for the for the muscular system, we're going to start with the head and neck. Notice we have some models that are similar to the ones that we have in the classroom. This is basically the one that is on one of our torsos. There's a couple other nice ones. So again, that's the one from our torso. Here's the one that kind of stands alone. Perfect. Excellent, as we can see that here. And for instance, we can highlight as we go through the muscles, the first muscle we are going to be responsible for is the masseter. So we can see that masseter. We can hear them say the masseter. We can do this. We can quiz ourselves. We talked about all of those things. The other thing that is going to be really useful for you, and I need to get rid of my annotations thing there, is up here at the top is animations. These are 3D animations of the muscles, and let's take a look at that masseter, because we're going to actually do this together.
The origins of masseter are from the medial surface of the maxilla and the medial surface and inferior border of the zygomatic arch. The insertions are to the lateral surfaces of the angle and ramus of the mandible. Masseter closes the jaw. It is innervated by the mandibular branch of the trigeminal nerve.
Now notice if you've looked at your study guide for the muscles, we have simplified those a little bit. We are just going to use the zygomatic arch as its origin and the mandibular angle as its insertion, which are the bone features you are responsible for. Notice the other thing too, the correct terminology is to elevate the jaw, not close the jaw. But again, it's still, what I like about this is that it isolates the individual muscles and shows you the individual muscles that you can study and look at that way. If you've looked online, one of the things that we are going to do as well, and again, I think I have to switch my share view to be able to do this. Perfect. As I've actually given you some drawings. So one of the things that you're going to be responsible for doing, and this is something that we'll start to do together, but I don't know how much of it we're going to be able to get to. As we just saw, you are going to be responsible for knowing the origins and insertions of muscles. And one of the good ways to do that is by drawing them. So I'm going to cheat and use my solid circles here to do this. As we just learned, the origin of the masseter is on the zygomatic arch. So I can take that and put that there on the zygomatic arch. Origins by convention, and again, that means the rule that people have made up, are red in color, whereas insertions are blue, and the insertion is on the mandibular angle. The other thing that is nice about this, if you take the time to draw the origins and the insertions, is the other thing that it helps you with is to understand the actions. Because as we'll talk about when we come back from our break and we start into the lab portion, when a muscle contracts, it pulls the insertion towards the origin. So in this case, when this muscle contracts, it would pull the mandible straight up, it would elevate the mandible. And so we can see that in its action there. So we're actually going to take the time to do that as well. All right. Back to this. So that is everything that I want to do for a lecture standpoint, but I do want to at least introduce you to how to work on some of these things for the lab. Normally in the classroom, we would have the opportunity to go through all of it together, but I do want to spend some time working on it a little bit together to make sure you at least know what is going to be responsible for you and to help you to move forward for that. But I went crazy over where we normally take a break. And again, I've provided you with a tremendous amount of information. So let's go ahead and take a break, and we'll go ahead and take a little bit of a longer break. Let's go ahead and go from this point, 12 or 13 minutes. We'll go to 2:35, and at 2:35, we'll come back. And at 2:35, we will start the lab portion of this. For that, I would encourage you to have that printout of the skull pictures that we just looked at, the bone pictures, have a red and blue pen to follow along. And then I would also encourage you to either have your textbook or an atlas so you can be looking at these muscles as we go through it as well. So we will go ahead, take our last break, come back and start working on the lab stuff. And I'll remind myself to start the recording again. And we'll do that, like I said, at 2:35. All right, any questions before we take our last break?
I do have a question about the toxins.
Yes.
So those toxins only affect parts of the body that use the acetylcholine as the neurotransmitter, right?
That is correct. But here's the one problem with that. Acetylcholine is the most common neurotransmitter we use in our peripheral nervous system. So that means all of our skeletal muscle uses acetylcholine, and many of the other effectors, especially for our parasympathetic nervous system, use acetylcholine as well. So you'll notice one of the things that they do with Botox, for instance, if you don't like the unsightliness of sweating, right? They will inject Botox into the sweat glands underneath your arms, and that can paralyze the release of sweat in those. Sure, you're now going to overheat as a result of that, but as long as your shirt's dry, that's all that matters. Or there's plenty of other places where they will use it as well. So yeah, acetylcholine, yes, the good news is it only affects acetylcholine. The bad news is acetylcholine is our most common neurotransmitter.
Okay, thanks.
Yep. Any other questions? All right, excellent. I will meet you back here at 2:35, and we will talk about how to be successful with the lab component. So, and obviously the last thing you'll need is your study guide of all the muscles that have the muscles, the origins, and the insertions, so that list. So make sure you have that as well. So those are the things you need. Bone pictures, muscle pictures, red and blue pen, and an atlas or your textbook. All right, see you in 10 minutes. All righty. A quick show of hands if you can hear me. Excellent. All right. That should hopefully be enough to mean that everybody is able to. All right, that's good. All right, so like I said, that is everything that we wanted to do for a lecture standpoint. As I mentioned, the good news is that the other two steps, the contractile cycle and the excitation contraction coupling, are not nearly as elaborate as the first step is. So actually, we will get through both of them in our next lecture. And barely remember what day is today. Today is Tuesday. So that means Thursday. Excellent. So Thursday, we will have the opportunity to go through those and finish that process. So we'll be done with that. So as scary as today was, it does definitely get better. However, the other big important thing that we need to focus on is on the anatomy. So hopefully what you have in front of you are those resources. And while I told you to get them, I forgot to get one of them for myself. So let me sneak over to the module and get the study guide for this. As you look at the study guide for this section of the muscular system, there's obviously some histology, and that we actually got a chance to go over in class, so hopefully that will work. But the other thing we need to do is the origins and insertions handout that we have, and then the muscle anatomy. Perfect. Now, when you look at that list that is in front of you, you have that list. That is the list in its entirety of the muscles you are responsible for. As I mentioned, there are over 600 named muscles in the body. So as you can see, this list is far less than 600. So it's a very relatively reasonable list that you have to know. And for all of the muscles that are on this list, you also need to know all of their actions. For a muscle like the masseter, that's pretty simple and straightforward. It has one action, it does one thing, it elevates your mandible. Other muscles on this list have as many as 2, 3, or even 4 actions. If a muscle has 4 actions, you are going to be responsible for identifying all 4 of those actions. So you are responsible for all that information and using the information appropriately, the right, correct anatomical terms. Now, there are 2 things about this that will help you. First, when you're talking about them, remember when we talked about motions, there's 2 ways you can describe them. For instance, if a muscle flexes my elbow, I can say it that way, that it flexes my elbow. I can say the joint where it has the effect, flex the elbow. Or I can say the part of the body that it moves. I could say flex the forearm. Flex the forearm and flex the elbow mean the exact same thing. All right, that's where you're making you do the curls for the girls so you can get the nice big guns when you flex your forearm or when you flex your elbow. It's the exact same action. So that's something that'll help you. The other thing that will help you on this, on the exam, is on the lab exam, whether it's online or whether it's in person, I'm going to show you a muscle. And on that muscle, I could ask one of those 2 questions, identify the muscle or identify the action. But I will not just say identify the actions. I will be precise. Identify the action of the muscle, or the 2 actions of the muscle, or the 3 actions of the muscle, or the 4 actions of the muscle. So if I point at a muscle on the head and ask you to identify the 2 actions of that muscle, well, then you know it's not the masseter, because as we just finished talking about, the masseter only has 1 action. So the nice thing about it is if you start thinking of in terms of which ones have 1, which ones have 2, which ones have 3, which ones have 4, once again, the wording of the question will help you to be successful on the exams. Now, as I mentioned also, as you look at that list, you need to know all the muscles on the list and all of the actions. But notice some of the muscles on the list are bold. Those ones that are bolded, and again, the masseter is one of those, and that means you are also responsible for the origin and the insertion. So for that muscle, there's 4 things you need to know. You need to know its name, you need to know its origin, you need to know its insertion, you need to know its action. Notice as you look further down the list, we have a muscle like the Orbicularis Oris. Notice for origin and insertion, it has dotted lines. Does that mean that the Orbicularis Oris doesn't have origins and insertions? No, of course it has origins and insertions. What it means is you're not responsible for it on the exam. So you are only responsible for the origins and insertions that are on this list. You're only responsible for the origins and insertions of the bold muscles. Now, there's one more important thing to know when we are talking about this, and we'll get to the muscles and everything else in a second. But the other big piece of information we need to talk about is what we call a muscle group. A muscle group, as the name would seem to indicate, is 2 or more muscles, no groups of 1. But there are 2 keys to the definition of a muscle group. The first key to the definition of a muscle group is the muscles must have similar functions. Notice the key word there. I did not say identical. They just have to have similar functions. But the other thing is that the muscles must, let's put that in capitals, share at least 1 attachment point. And of course, by attachment point, I mean origin and or insertion. So just because two muscles have similar functions don't mean they're a muscle group. They also must share at least one attachment point. They either must all start from the same place or all end at the same place. And in a couple instances, they will do both. All right. If you look on those lists, there are some muscle groups where you just need to know the muscle group. A great example of it, as you look on that list on the first page, is the Zygomaticus. Notice under Zygomaticus, it actually says in parentheses, muscle group. The Zygomaticus is actually made up of two muscles, the Zygomaticus major and the Zygomaticus minor. But guess what? You don't have to know that. If, like the Zygomaticus or the scalenes that is above that, I just have the group, then that's all you need to know. You just need to know those groups collectively, where they originate, where they insert, what they do. You do not need to know the individual names. However, if, for instance, you go to, let's say, page 4, notice on page 4, there is a muscle group known as the hamstrings. The hamstrings is comprised of 3 muscles. And as you look on your list, you'll see those 3 muscles are the biceps femoris, the semimembranosus, and the semitendinosus. In that case, because they're listed, if listed, you must know them collectively. The collective group name, like the hamstrings, but you must also know the individual muscles and all of their information. And again, it's all right there in front of you. The quadriceps femoris is a muscle group, and you need to know all 4 of those muscles. The hamstring is a muscle group. You need to know all 4 of those muscles, right? However, like I said, the rhomboids on page 2 is a muscle group that you don't need to know the individual muscles. So I've tried to make it as straightforward as possible by either letting you know that you just need to know them collectively as a muscle group, and all the muscle groups should say muscle groups on them. There's one I'm peeking for. Yep, no, even there, it's there too. Excellent. And then the 2 that you need to know the individual muscles are the hamstrings, the quadriceps femoris, and then there's one other muscle group you'll need to know as well, and we'll get to that when we get to that. All right. So those are the things you need to know for these things. Now, one of the things that I find useful for this is flashcards. I know not everybody likes flashcards, but I find flashcards to be very, very useful for studying this. However, the key to a flashcard is you have to use the flashcard correctly. Too often when people do this, what I see is they put the name of a muscle on one side of the flashcard, and on the other side of the flashcard, they write out the origin, they write out the insertion, they write out the actions. And what they do to study these flashcards is they stare all day long at the name and try to think of the other 3 pieces of information. But these 3 pieces of information you really need to know and understand independently. As I mentioned, whether this is a test on the station-based, where we're going to be in the classroom and do it, or whether we are going to be online doing this, I will show you pictures of bones. So I will show you an origin and ask you what muscle originates there, or I'll show you a bone feature and ask you what muscle inserts there. What I find one of the easiest ways to make a successful flashcard that helps you to be successful is to take a flashcard and fold it in half. If you take that flashcard and fold it in half, essentially what you have is a 4-sided flashcard. What you can do on one is write the name, or what I find works even better, take one of the pictures of the muscle and put it on there. If you look online, hold on, there you go. If you look online, you'll see there are some unlabeled muscle pictures that are available for you online that you can look at. So that is something that you can start with. And on the other side, you can then put the origin. right? And again, make sure you know it's the origin. So O equals origin, because sometimes something that is the origin for one muscle will be the insertion for the other. On the back side, on one half, you put that in, you can put an insertion. And then on the fourth half, no, 4 halves doesn't work, but you get the idea, you can put the actions. Then what you can do is you can take these flashcards and fold them. So you can look at just one of these pieces of information. So if, again, we did that with the masseter, because that's the only one we've looked at so far. If I looked at the action, I would see the action is elevate the mandible. The one action is elevate the mandible. So then I would think, all right, what is the muscle that we've learned that has one action, and that action is to elevate the mandible? Or I could look at the insertion, and I would see, all right, I would see the insertion is the mandibular angle. And I could try to think, all right, what muscle inserts into the mandibular angle? Or I could see the origin is the zygomatic arch, and I could try to think of what that is. Or I can look at the picture of it or the name of it and try to think of those things. By separating the information and using each one as a way of trying to trigger the memory and trigger the understanding, hopefully that can help you to be successful with that. All right. Questions on any of that? Elena, what did you say no for? What was the question that I asked that you're saying no? All right. So this is something that can help you. And then again, we'll go through some other things that will help as well. However, before we get to the origins and insertions, the one last thing that we need to make sure we understand is, again, the first thing really we need to do is identifying these muscles. When we identify the muscles, there are basically 3 ways that we identify muscles, right? With over 600 muscles in the body, we obviously had to come up with some rules of how we name them. And if we've learned anything from anatomists, do they always stay 100% to these rules? No, of course not. Anatomists are jerks, but it's going to be close. And so when it comes to the 3 ways we identify the muscles, there's 3 primary ways. The first is the location of the muscle, right? Your bicep brachia is in your brachial region, your upper arm, whereas your bicep femoris is in your femoral or leg region. So often muscles are given names based on their locations. Second way we identify muscles is by their actions, right? A flexor flexes, an extensor extends, an abductor abducts. So often muscles are given names, elevators elevate. So we're given muscles names by their location, by their action. And the third way that they will identify muscles is by the muscle shape. This helps us 2 ways. The shapes are one of the ways that they name the muscles, so that helps us to understand that. But the other thing that is helpful is that it helps us to, knowing the shape, as we talked about, will also help us to understand the action, how it is going to move. All right, questions on that? All right, excellent. So with that in mind, let's talk about, whoops, wrong button. The shapes of the muscles and the anatomy itself. As we've talked about, we need to be able to identify the muscles based on their actions, based on their location, based on their shape. And we need to know origins and insertions. As we talked about by rule, origins are always represented with red, and insertions are always represented by blue. So as you look in your atlas, or if you look in your textbook, you'll see places where you'll see them represented that way. And that is just the rule that they have always used for these things. In general, and again, if you think about it, and let's cheat and draw a couple bones. Here is one bone, here is the other bone. And then of course, we are going to have a muscle that connects them. When we have a muscle that connects bones, when I have a muscle that connects to the bones, it almost always has to cross a joint. If it doesn't cross a joint, it really can't have an effect. If we had a muscle that just went from one part of this bone to another part of that bone, when it contracts, it wouldn't have any function, it wouldn't do anything. So typically a muscle will cross at least one joint, and every joint it crosses, it has the ability to affect. So that's the other advantage of drawing these things. We can see the joint it crosses, and then that can help us to understand the effect. The other thing to understand is when this muscle contracts, this bone doesn't come up this way, and this bone doesn't come up this way, and they meet in the middle. That's not how it works. Typically, when we contract a muscle, what happens is one side stays stationary, does not move, where the other side moves. So typically, the origin, and again, that origin is going to be in red, is the end that stays, let's make that a solid circle. The origin is the part that stays stable, is the fixed end, does not move, whereas the insertion is going to be on the movable bone, the one that does move. All right. Now, there are some rules. That is the primary way we identify origins and insertions, although, again, like all things, there can be some funkiness to it. In general, and of course here, the key word is, of course, typically. Typically, origins are more proximal or closer to the midline. Typically, origins are bigger, they're broader, and insertions are more narrow. And again, keyword typical, there are going to be some differences in that. But here's the other big important thing we need to know about origins. Origins, some muscles have multiple bodies or multiple heads. In fact, that's what bicep brachia means. Bicep brachia means two heads in the brachial region, in the arm region. When a muscle has multiple heads, typically it has multiple origins. So again, if we think of this muscle, and let's cheat and we'll draw it solid and we'll use pink. Oh, this isn't going to be, oh, okay, this is going to be ugly, but it'll work. Typically, what happens with a muscle, and again, obviously this is exaggerated here, we have an origin for this head, we have an origin for this head, and they are going to come together to a single insertion. Now, when we have a muscle like the bicep brachia, then if you think about it, there are going to be three attachment points you're going to need to know. The origin of this belly and the origin of this belly. And on the exam, it makes a difference. If I put a tag, a big number 1, on this belly during the exam, and I ask you to identify the origin of this belly, of this muscle, I don't want both. I just want the one for the belly I indicated. And I will give you two key words that are going to be the very important bells and alarms go off when you see them. Oops, helps if I spell it correctly. The two key words you will see on the exam that lets you know there's more information involved than you think is going to be that magical term, be specific. If you see be specific, that doesn't mean give me all of the origins. That just says, give me the origin of this one belly. These two bellies have different names, right? So you need to give me, if I ask you for the name of this one and I ask you to be specific, you need to give me that specific name. Don't give me both names, right? Just give me the specific name. And again, when it comes to insertion, notice they have one insertion. They're also going to have one action. So the two things that will be specific will either be names or origins. So if you see those two magic words, be specific, that's what I'm looking for. It means I want a specific name or specific origin. If you give me both, you are showing me knowledge. You're showing me more knowledge than I've asked for, but you haven't been specific, and you will not get full credit for that. So make sure you are specific for that. And again, those are the two magical words that should allow the warning bells to go off. Be specific. All righty. Questions on that? Excellent. We already talked about muscle groups, so I don't have to go over that again. So excellent. So that is that. The last thing we need to do is talk about, again, our names. Our names, as we talked about, are based on location, based on action, and as we talked about, based on anatomy, their shape. Now, we know muscle cells are all bundled together inside of a fascicle. So inside of a fascicle, we have a bunch of muscle cells, right? That muscle cell and 14 of his closest buddies are all bundled together, parallel to each other inside of a fascicle. This is important because if cell 1, cell 2, or cell 3 fires, we're all going to get the same action. We are going to get the insertion moving towards the origin. So again, in all of these cases, this is going to get shorter and the muscle is going to move that way. So all muscle fibers in a fascicle are parallel. However, when we make a muscle cell, pardon me, when we make a muscle the organ, the fascicles don't have to be parallel. Now again, I haven't figured out how to rotate these things yet, so I'm just going to do it perpendicular. So notice these two fascicles are not parallel. Again, the muscle cells inside of them are going to be parallel, but the fascicles themselves do not have to be. And the reason this is important is because when we contract this fascicle, it's going to pull this bone this way. When we control this one, it's going to pull this bone that way. So basically, the relationship of the fascicles determines the shape, and the shape determines the function. Let's just see the examples. I think it's easier if we make some sense of this. So again, all the muscle cells in a fascicle are parallel, but all the fascicles are not. And the organization of the fascicle determines the muscle shape. There are 4 major shapes. Here we see the pretty pictures that look at them. And here is our first. Our first shape are what are called parallel muscles. As the name indicates, with a parallel muscle, all the fascicles are parallel to each other. And again, the significance of them being parallel is that if one fascicle contracts or a separate fascicle contracts or a third fascicle contracts, you get the exact same action. Most of the muscles in our body are parallel muscles. And one of the reasons this is significant is like we talked about, right? Again, this is a lot more fun when we're in person in class because I can be more light about this because this is exhausted material and we've been talking for a long time. But my go-to joke is the whole reason we contract muscles is to woo women, right? So you do, like you said, you'll go to the gym, you see all the boys there in the gym doing all those big curls, right? You do the curls to the girls so that they can show off their big guns because that's what happens with a parallel muscle. That big superficial muscle that is the bicep brachia is a parallel muscle. They all pull the same direction. And if you think about it, when the muscle contracts, not only does it get shorter, but as it gets shorter, it also thickens. It gets fatter as well. So as this muscle contracts, it balls up in the center. And so we get these big ball-shaped muscles as it's contracting. And those are those big, massive muscles that stick out when we flex. So here we have that parallel muscle with all the parallel fascicles, tendon at both ends, and as it gets shorter, it also gets fatter. This is great for most of the parts of the body. Like I said, you get the curls for the girls, or you get those nice big huge calf muscles from wearing your high heels all day long, or all of those types of things. We have these big parallel muscles. But there's some areas where that parallel muscle isn't going to be as useful. One of those is the chest region. Your bicep brachia is this big, huge fan-shaped muscle like we see here in the picture. Notice it has one central point where it comes together, but then it has this big, broad base to it. It has kind of a fan-like appearance. And so notice all of these fascicles are at different orientations. All right? The advantage of this is flexibility of movement. right? My bicep brachia, I just need to flex my elbow. That's pretty much all it does. It moves my arm in one direction. But when it comes to my chest and affecting my shoulder, I want a lot more flexibility. I want a lot more action of the shoulder. And again, if you think of it in terms of working out, because many of you have worked out before or know people who work out or like to work out, if your goal is to increase the size of your chest muscle, typically you do a bench press, but do you always necessarily do it on a bench that is parallel to the ground? Or do sometimes you incline or sometimes you decline so that you're moving at a different angle, right? And that's what that big, huge convergent muscle allows you to do. It allows you for a lot of different angles of motions that we can get with that converging muscle there. Big fan shape, single attachment point, and again, it allows for flexibility of movement. This is really useful for places like the chest or our hip, where we want big range of motion with those ball and socket joints. However, we look down at our forearm, and not everybody wants big, huge Popeye forearms. I don't know how many of you get that reference, but back in ancient times, there was a cartoon called Popeye. He was a sailor man, and there were three things about him. He had a horrible speech impediment. He dated olive oil, and he loved spinach. He'd eat massive amounts of spinach and made it super, super strong. Aside from the fact that he smoked a pipe, the other big characteristic that really made Popeye stand out is he had these big, huge, massive forearms. Right? He had forearms like thigh muscles. They were big, huge, massive forearms. And as you look down at your forearm, most of you don't have that. The reason you don't have that is because you don't want that. You don't want big, huge, massive muscles in your forearm. But what you do want is a lot of strength. And that's where a pennate muscle comes in. Again, I'm somewhat limited on my drawing here, but we'll cheat. With our pennate muscle, what we want is we want our tendon, and we want to connect our fascicles to it in a way that provides a lot of strength in a small place of space. This is normally where, when we're sitting in the classroom, we talk about playing tug of war again, where I have all of you on one side and all of me on the other. And if all of you are on one side and I'm just on the other side by myself, you guys are going to beat me at tug of war all the time. However, to do that, all of you need to be able to get a hand on the rope, right? So that's what we want to have. We want to have a lot of hands on the rope. And that's what happens with a pennate muscle. In a pennate muscle, all of the fascicles are parallel to each other, but they're not parallel to the tendon. And what this allows us to do is get a large number of fascicles onto the tendon in a very small space. So notice here we have a large number of fascicles, all parallel to each other, connecting to a single tendon. That gets us lots of hands on that rope for tug of war. And what it allows us is to have a relatively small muscle that has a tremendous amount of strength. And that's what we want in small compartments, small areas like your forearm. In your forearm, you want to have small spaces with just a small amount of muscle cells in there, but you want a lot of strength. Now, there are three types of pennate muscles. There's the one that I just drew right there, which is what we call a unipennate, and that's what we see down here, unipennate. We have one tendon and one row of fascicles. Again, we have a large number of fascicles producing a lot of tension. They're all parallel on a single tendon. That is our unipennate. But we can also have a bipennate. With a bipennate, we still have a single tendon. And let's change colors here. And then we have parallel fascicles. But in this case, they're parallel on both sides. So we have those parallel on this side, and we have these parallel on this side. This muscle, the example you see here is the rectus femoris. It is one of the muscles of your leg. It's one of the most important muscles of your leg. It's a relatively small muscle, but because it has a massive amount of fascicles in it, it is an incredibly strong muscle. But if you think about it, it makes sense. Your leg is a big, huge chunk of meat, right? It's one of the biggest, largest, heaviest parts of your body. And to be able to swing that around requires big, powerful muscles. Well, what we have right here in the front on top of your leg is this big, not big, but is this really strong, super strong muscle, one of the strongest muscles of your body, this bipennate muscle that is the rectus femoris for moving that big, huge, massive piece of meat. And that is a bipennate. The third type, and I'll see if I can sneak that, nope, not gonna work, so I have to clear my drawing. The third type of pennate muscle is a multipennate. With a multipennate, what we have are two or more. And here, your deltoid, which is that big superficial muscle on the outside of your arm, is a good example of this. It has 3 tendons, but for simplicity, I will just draw 2. And on these, you can have either one line of parallel fascicles, or you can have 2 lines of parallel fascicles. But with a multipennate muscle, again, we get a lot of strength in a small space, right? If you think of your deltoid muscle, the deltoid muscle is the big, huge chunk of flesh on the outside of your arm. It has a tremendous amount of strength, allowing you to be able to raise your arms up and move your arms around in a relatively small space. And so again, these pennate muscles give you tremendous strength for a relatively small size muscle. The last shape, and one of the easiest to understand, is a circular muscle. And of course, how are the fascicles arranged in a circular muscle? In a circular fashion. That's pretty simple and straightforward. Now, when we have a circular muscle and that circular muscle contracts, what happens to the size of the lumen, right? Because remember, this space inside here is the lumen. And when we contract a circular muscle, what happens to the size of that lumen?
It's smaller.
It gets smaller, absolutely. So again, fascicles form concentric layers around an opening. And when it contracts, that opening gets smaller. All right. These are really important for openings. We have a circular muscle around our eye, circular muscle around our mouth, circular muscle around our anus, all these areas where we want to control the movement of things in and out. These form in smooth muscle as well. And in smooth muscle, this circular layer thickens where it actually can become a valve, where it can actually control the movement of things in and out, at which point we then call it a sphincter. So those sphincters are examples of thickened, large circular muscles that basically act as valves, controlling the movement of things like food and other structures through our body. All right, questions on that? All right, so like I said, there are lots of ways for you to go through this and try to make sense of it. Here are the pretty pictures from your textbook. Here's an anterior view. Here is a posterior view. Here is a side view of the body. And again, these pictures are right out of your textbooks. Your textbook has this. Your atlas has great pictures of these. Your lab manual has great pictures of these. So however you want to study these things. And of course, as we saw, the Master in A&P in that lab thing has this as well. So here, of course, as we talked about, is our masseter. As you're studying the masseter, as we mentioned, one of the things you're going to want to look at is our skull. We already did this, but apparently it's not still here, so I don't know if that means I'm gonna have to do it every single time when we come to it. But remember, as we talked about, the origin of the masseter, as it shows on the list, is our zygomatic arch. So I will draw that circle and put it there. Again, you can color it in when you're coloring in a little bit clearer. For simplicity, I'm trying to use circles here just because it's simpler. And the insertion is our mandibular angle. And as we talked about, when we contract a muscle, the insertion always goes towards the origin, and so it elevates the mandible. Again, you can experience this for yourself. This would be the kind of thing you use when you take a big bite of something, or if you're chewing something. If you take your hands and put your hands on the sides of your cheek and you clench your teeth, when you do that, you actually feel the masseter muscle in your jaw tensing. You can actually feel it tense and relax as you clench your jaw. And that's what we are looking at there. That is the masseter. So really, that's all I'm looking for you guys to understand and do. Know the origin, know the insertion, and that can help you with the action. And that's, of course, what's on our list. All right. Now, again, notice there can be muscles in similar locations, and that's one of the advantages of actually taking the time to draw the origins and insertions. Another muscle that plays an important role in our chewing or moving our jaw is the temporalis. Notice the temporalis is superior to the masseter, so they're not quite in the same location. And if we go back to our skull drawings, we can see here, and if we look at our study guide, we can see that indeed the origin of the temporalis is going to be that temporal fossa up here on your, above your zygomatic arch on the side of your temporal bone, that nice smooth part of the temporal bone. I made that a little big, but we can get the idea. I don't want it on the zygomatic arch, so let's do that. And the insertion is on the coracoid process. Now, again, I'm going to draw my circle here. Oops, I'm going to draw my circle here. Now, we have a little bit of an issue because if you really think about it, this drawing doesn't truly show where the end of that coracoid process is. We know it's kind of in here, so we can cheat. Let me move that out of the way and get rid of that. Maybe I'll just use a line instead. For that line, what you can do is you could color in just the part of it that you could see. Or what I like to do, because it makes sense to me, is I will draw a dotted line. That dotted line basically tells me that is where it is. It's just hidden by the other bones. How you draw this, there's no right answer. You're not turning these into me for a grade. What you are doing is making a study guide that can help you to learn this. In fact, you can cut these pictures out and put them on a flashcard, and that can help you to learn this material as well. So draw it, just do it however it is that makes sense to you. However, again, the advantage of drawing this is, again, to see that relationship. When a muscle contracts, the insertion is always pulled towards the origin. And notice, remember where our masseter, even though my pictures aren't here, we know that came straight up. We could see the joint it affects the mandible. This one affects the mandible as well. But notice this one pulls the jaw at an angle. It pulls the jaw up, but it also pulls the jaw back. So whereas the masseter has one action, how many actions does the temporalis have?
2.
2. One of them is to elevate the mandible or elevate the jaw. The other is to pull it back. And what's the fancy word for pulling back? Again, you should have your list in front of you, so it should just be a matter of reading it off. Oh, there you go. Someone typed it out. Retract. Excellent. To retract. So the temporalis has 2 actions. It elevates and retracts. All right? So again, notice taking the time to draw the origin insertion can sometimes help you to know what it's going to do. It shows you the joint we're crossing over. In this case, the joint is the jaw. And or what it's moving, the mandible, and we can see the direction it's going to move it. It's going to bring it up, and it's going to bring it back. All right. Now, there is something else that I encourage you to do. This is something we would do in the classroom to help us. I am a big proponent of mnemonics, tricks to help you to remember this information. All right. Now, with something like the masseter that has just one action, you don't necessarily need a mnemonic to help you. But other ones can be kind of tricky. And so what I like to try to do as I'm going through these things, and normally what I would actually have you do if we were in the classroom, I would actually make you come up with these on your own and you would actually demonstrate them for me in the classroom, is to try to come up with an everyday activity where, or maybe even not so everyday activity, where an activity that would use all of the actions of this muscle. So like for instance, being an older brother as I am, right? I often, growing up, would use my temporalis to bring my jaw up and bring my jaw back to give my sister what I would commonly refer to as the dork face, right? Whenever you're making that durr sound to let them know that they've done something stupid, to try to embarrass them, make them feel bad about themselves, which, as an older brother, you are required by law to do. that dur face that you make is that elevation and retraction of the mandible. All right, let's do one more because the next one's my favorite. Let's go back to the, come on, pretty pictures. Oh, is the next one my favorite? Uh-oh, I lost my pictures. Oh, yeah, it is. Next one is my favorite. Excellent. Favorite muscle is the sternocleidomastoid. All right, with the sternocleidomastoid, it is again one of those alphabet soup type words that you can be guaranteed you're going to have to spell on the exam. But like many of these alphabet soup type terms, there is everything's in the name. So when you look at the name of it, sterno refers to the sternum, clido refers to the clavicle, and mastoid refers to the mastoid process of the temporal bone. As it turns out, this muscle has 3 attachment points. And guess what those 3 attachment points are? How about the sternum, the clavicle, and the mastoid process? Now, when we look at this pretty picture from your textbook, this one where we see all the superficial muscles and such, you don't necessarily get a chance to see all of it. However, if we peel away some of the other muscles, what we can see deep inside here is the sternocleidomastoid. And we see that it indeed has 3 attachment points. Notice the origin is twofold here on the sternum where it attaches to the manubrium. And notice there is a little bit of gap. It's not continuous, so it doesn't continue all the way onto the clavicle. It's 2 separate origins. It originates from the manubrium and it originates from the superior surface of the clavicle. And then the muscle inserts into that big bump behind your ear, the mastoid process of the temporal bone. So those are our 3 attachment points, 2 origins and 1 insertion, the sternocleidomastoid. All right, questions on that? So notice if we go back to our drawing, and I think I can slide it over. Excellent. Here we have our sternocleidomastoid bones. So again, we want to draw our origins first. So we're going to do that in red. We know part of it is out here on the manubrium. Part of it is over here on the clavicle. And then our insertion is on the mastoid process. Notice we see a nice mastoid process over here on the turn one. Do we really see it over here? Really? This picture doesn't really show it. So what I do, again, because I like to cheat and make sense to what makes sense to me, I kind of draw it back here, kind of where it would be, and then I can write next to it, it's the mastoid, to just remind me that it is back there. All right. Now that we've drawn those, as we know, when we contract a muscle, we are going to pull the insertion towards the origin. Most of the time. That was that pesky word, typical, that we saw. Remember also, all these muscles, we're just drawing one of them, but remember these are paired muscles. We have them on both sides. And for this, I'm going to turn on my camera. So let me turn on the light so you guys can see me. You'll see that I have not trimmed my beard or shaved in weeks now, so I'm all scruffy and everything. That light. There we go, and let's take a look. That work? You got me? All right, excellent. So if you think about it, the sternocleidomastoid, his job is to bring the mastoid process down towards the clavicle and the sternum. And you have 2 of them. When you contract them both at the same time, it brings the head down. It flexes the head, or flexes the cervical vertebrae, the neck, whichever you want to think of it. In fact, the sternocleidomastoid is called the prayer muscle because of that. When you contract them both, it brings the head down. However, if you contract just one of them, again, our goal is to bring the mastoid process towards the sternum and the clavicle. So what happens is it brings it down, and as it brings it down, it rotates the head. So notice if you just contract one, it rotates the head. So both flexes the head, one rotates the head. You guys got that okay? With me so far? Excellent, a couple of yeses, perfect. Now, here's the challenge. This is not the only muscle that moves the head. I have other muscles that can control my head. And this is where muscles get a little bit tricky. One of the things we can sometimes do is use other muscles to stabilize my head. So that when I contract the sternocleidomastoid, instead of my head going down, what I can do is hold my head in place. And if I do that, instead of the insertion going towards the origin goes towards the insertion. So if you think about it, if I stabilize my head and refuse to move my head, when I contract this muscle, instead of my head coming down, my sternum and therefore my rib cage comes up, like when I'm taking that big inhalation. So if you notice, there are three actions for this muscle. We can flex the head, we can rotate the head, and we can elevate the rib cage, like an inhalation. Now, like I said, I encourage students to come up with common or not so common activities that help them to remember this. I've been doing this since the beginning of my grading and teaching. And so this was probably 10 plus years ago. I had a student. So again, you think about how life was, it's very different now than it was 10 years ago. And I've gotten some great examples. Usually people come up, one of my favorite ones that people have come up with, like for instance, if you're at church, right? When you're at church, you're bowing your head, of course you're in church, but of course you also have to talk about the people that you're, gossip about the people. So you're kind of rotating your head side to side so you can talk and, you're surprised by something you see or something like this. But about 10 plus years ago, before it was legal, I had a student, very quiet student, sat in the back of the class. And when she got up to demonstrate, she demonstrated what she referred to as a bong rip, right? For her bong rip, flex the head, elevate the rib cage, and rotate the head. So there you go. You put those three actions together into a bong rip, and you can remember all three actions of the sternocleidomastoid. All right, questions on that? All right, excellent. Well, heck, after talking for 4 hours, doesn't hurt to be a little hilarious. What about the Zygomaticus? We will get to the Zygomaticus. It is definitely on our list, but I was just trying to go through order. So that was the third one on our list. All right, so we will get to the Zygomaticus. Any questions on the sternocleidomastoid? Now, now that you've seen me pretend to take a rip from a bong, there is no reason for you guys not to remember the three actions of the sternocleidomastoid. I expect no one to get that wrong on the exam, but more importantly, it should give you an idea of how you can do this for yourself. For yourself, try to come up with things like the Durface, like the bong rip, that help you to remember what the activities are of these. And again, I'm not going to ask that on the exam. I'm not going to ask what muscle allows you to do a bong rip, but I am going to point at the sternocleidomastoid and ask you what its three actions are. And if remembering that bong rip helps you to remember those three actions, then that is going to allow you to be successful on the exam. All right, let's do the Zygomaticus, because if you look, there's only two, well, actually we have one more before that, the scalene. So let's go back and do that one as well. Hold on. There. If I remember correctly, come on. So I have to move all this stuff on my screen. There we go. There we go. And here we go. All right, there's the picture for that, but let's go back to this first. So any questions on the sternocleidomastoid before we move forward? All right, excellent. Here is the sternocleidomastoid. And again, notice here they've gotten rid of the pesky face of our skull, so we can clearly see the mastoid process and how it connects. And what's nice about this is we get to see a muscle group that is right next to it, deep to it, and that muscle group is known as the scalenes. Now the scalenes happens to be made up of 3 individual muscles, the middle scalene, the anterior scalene, and the posterior scalene. And do you need to know the names of those 3 individual muscles?
No?
Nope. You just need to know them collectively as the scalenes. So if we get rid of, get rid of the picture, here we see the scalenes. And notice here, and I'm not going to bother to go and draw it on the skull. Well, actually, I guess we can do that. If we look, now that we see where the scalenes is located, we can see what the origin and the insertion of them are. Notice the origin is here on the transverse processes of C2, C3, C4, C5, C6, and C7. So those transverse processes on those cervical vertebrae. And notice it inserts into the anterior and lateral part of ribs 1 and 2. Notice the sternocleidomastoid came more anterior to the manubrium, to the clavicle. These are going more lateral. And we see the differences in their actions because of the differences of their attachments. Notice this one rotated the head, and the scalenes rotates the head. This one flexes the head. This one also flexes. So it has 2 shared actions. But the sternocleidomastoid, because of the twist of this all the way to the sternum, or the manubrium of the sternum, rotates the head. Whereas notice these are much more lateral. So again, we'll go back to the video one last time. If you think of this with the sternocleidomastoid and the scalenes, both flex the head, right? Both have the ability to elevate the rib cage. So they both can do that. But the sternocleidomastoid is going to rotate. The scalenes, because it's more lateral, it is going to laterally flex the head. So laterally flex for the scalenes, rotate for the sternocleidomastoid. All right, so I always think of this one as like, this is one of those dog muscles. When you say something to the dog and the dog's paying attention to you, it gives you that little twist of the head, right? That little lateral flexion of the head. That's what that scalenes is doing. So it's flexing the head. It is elevating the rib cage. These are muscles that help us take that exaggerated breath, but the scalenes is going to laterally flex the head. All right. And I'm not going to keep demonstrating all of these so you guys can make fun of me, but I just want to make sure you guys are understanding what you're responsible for on these. All right? Questions on that? So that's going to be the goal. You have the list in front of you. You have pictures like this picture in your textbook where you can see, right? Oh, and since it was asked, and I tell you what, since it does have an origin and insertion, we will do the Zygomaticus as well, and because it was asked. The Zygomaticus, notice, and again, let's go to our spotlight here, is again a muscle group made up of 2 muscles, the Zygomaticus major and the Zygomaticus minor. However, do we need to know the names of the individual muscles? No, we just need to know them collectively as a muscle group. Notice what's unique about, well, not unique, but what is, well, it is unique-ish about the Zygomaticus muscle. It has an origin on a bone. After all, most skeletal muscle connects to bones. And notice it connects here to the zygomatic bone. Notice not quite out here quite as much on the zygomatic arch, more just the zygomatic bone, right? So we can see that it's kind of here out here on the zygomatic bone. That's that apple of your cheek, if you think of it in terms of that. But notice where it inserts. It doesn't actually insert into a bone. Instead, it inserts into the muscle and the skin at the lateral aspect of the oral cavity. So when this muscle contracts, the lateral aspect of the mouth is brought up and back for that big smile. So again, it elevates the lateral edge of the mouth, or what we commonly think of as smiling. It's one of the primary smile muscles. Brings it up for that. All right, so that's our goal. Take the time to start working through these. The good news is that's it. Those are the only muscles of the head. And notice also, as you look on this list, the same way we tried to break up the skeleton into different parts so that it would be easier to digest the material, We talked about the bones of the head and then the bones of the vertebral column and even axial and appendicular and stuff like that. We're doing the same, we're doing the same thing here with the muscles. The muscles that move the head, the muscles that move the trunk, the muscles that move the arm, move the leg. So we've done those. We've hit all the ones with the origins and insertions, talked about how you would draw them, where you would draw them, what they do. And so now for the rest of them, you just need to know the muscle and the actions. And again, your book's got great lists of these. I think what I will do is each day, depending on how long the lecture takes, we will try to go through at least the ones that have the origin insertions. So for Thursday, we'll try to go through the origins and the insertions of the trunk. There aren't too many of those. We'll also talk about some of the trickier ones like the levator ani and the diaphragm. So we'll go through those lists. I don't think any of the other muscles, actually there is just one muscle that I wanted to point out on this list. So let's jump ahead to it. Oh, we'll get to that. It is the occipital frontalis. Excellent. The occipital frontalis, as the name would indicate, is a two-bellied muscle. One muscle is in the occipital region, the other is in the frontal region. So we have this occipital frontalis here that has two bellies. So this is another one on the exam where I would have a tag right here and say, identify the muscle, be specific. Or I put it here, identify the muscle, be specific. All right, and the answer to those two questions are different. This is the frontal belly of the occipital frontalis. This is the occipital belly of the occipital frontalis. Now, if you're looking at your study guide, there's one other thing that I want to point out to you. If you look at the bottom, there is an asterisk and it says should also be able to identify. These are non-muscular things. And with non-muscular things, I will use the keyword on the exam, structure. So I'll ask you to identify the structure. Oops, no, go back. That's what I want. And again, I'm not putting this here by mistake. That is where I want that. Hold on. This particular structure, notice, is a structure that connects the two bellies together. This is a big, broad, flat tendon. And if you remember, we gave a big special word to a big, broad, flat tendon, and that word is aponeurosis. So aponeurosis is just a big, broad, flat tendon. This big, broad, flat tendon on the top of your head is the epicranial, because it's on top of your head, aponeurosis, and it connects these two bellies together. You may not have thought of it in these terms, but you're aware of it. As we've talked about the occipital frontalis, and as you can see on the study guide in front of you, it elevates the eyebrows. So if you put your hand on top of your head and elevate your eyebrows, what you can actually feel is you can feel your whole top of your head moving. It isn't just your eyebrows that go up. The whole scalp of your head moves. And that's because your scalp is underneath, right? Underneath the scalp, I should say, is that epicranial aponeurosis that connects the two together. So your hand should jiggle back and forth as you elevate your eyebrows. And I spared you having to look at me do that. So there you go. You have that epicranial aponeurosis. That is a structure. And so if you look at the bottom of each page of lists, there are additional structures you could be responsible for. So that's the game plan. That's what's going to be expected of you on the exam. Using pictures like this, using pictures like the pictures of models. And again, if we talked, as we talked about, hold on, let me stop sharing this for a second so I can go back. Where was that? Ah, here, perfect. Excellent. So this is our module section. This is, again, where your unit review drops it. These are where you drop off your unit reviews, your physio-X exercises. This is where the Dropbox for all of those things are. Your lecture slides, your histology, your anatomy, the pictures, the unlabeled muscle pictures. Oh, one other thing that I want to point out, There is bodybuilder pictures on here. There are, these muscles obviously are the same muscles that bodybuilders have, but as you can see, when the bodybuilders exaggerate these muscles, they tend to get really large and can be displaced. When we do this exam in person, if we have an in-person exam for this, one of your stations will have bodybuilder pictures. As we go through the muscles, we'll look at some bodybuilder pictures to see these. You do need to be able to recognize these on a bodybuilder. So I've given you a nice chart here that helps you to be able to do that. So you have some great tools there that are here. So you have all of those things here that are here. Here are where those communication of the neuromuscular junction lecture pictures are, but I'm not going to bother you with that. But what I did want to remind you of is that lab study tools that, you know, Cosumnes River College has that virtual anatomy lab. And if we go to that virtual anatomy lab and we scroll down to the muscular system, right, and we go to things like the, I wonder if the torso would have head on it. Let's find out. There you go. Lo and behold, here we see the masseter. Here we see the zygomaticus, right? Here, up here where the temporalis is cut off, but notice they put a little bit of it on here where we can see it. The Orbicularis oculi, the frontalis, which is the frontal bone, frontal muscle of the occipital frontalis, Zygomaticus, Orbicularis Oris, Masseter, Buccinator, sternocleidomastoid. Perfect. All of these muscles are muscles that are on your list that you're responsible for. Now, for every picture in here, will all of them be pictures? Will all the muscles that are listed be necessarily ones that are on your list? No, of course not. So just ignore the ones that aren't, right? The splenius capitis is not one of the muscles you're responsible for, so feel free to ignore it. But there are plenty of others that are on here that are really good. These are pictures of the models. Pictures of the models are something that either will definitely be on the exam, or if we get to do this in person, it will be a station-based exam, and you will be responsible for the models, right? This is exactly the torso that we have, and these torso models are amazing. They have great material on them. They're really, really excellent. I know I bag on the model makers a lot, but the chests of these things, the walls of these things are really, really excellent, where you can actually see the different layers of the abdominal muscles, even though they're not labeled on this particular picture. So there's some really, really great things on this. So take advantage of this site. So again, when you go to the modules, the lab study area is there. We looked at the modified mastering A&P. There's a lot of great resources there. Make sure you're taking advantage of the materials that you have to help you to be successful on this exam. All right, I can walk you through the lecture, but the lab part is just as important. And again, you're going to have the more of the onus is on you to make sure that you're getting the material and you're being successful with it. All right. Any questions on that or anything else? Now you have the game plan. You know what you need to do. You know how to do it and how to be successful. So let's go back to that. So we have that. And so we'll do more of that. We'll try to, like I said, we'll try to at least make sure we get all the ones that are of the origins and insertions, or ones like this that have something unique about them that you need to know. And we'll hit all the structures as well. So we'll go through all of them. We won't be able to hit everything on the list, but we'll try to highlight the key ones. But you are responsible for everything on the list and everything we talked about in the lecture. All right. Any other questions? All right, well, thankfully, that is all that I have for today. So that is all that I have to cover. Hopefully that is more than enough information to keep you busy over the next couple of days. And again, like I've said many times, and this is true in the classroom as well as it is true here. Obviously, I'm talking for a very long time, covering a tremendous amount of information. It's one of the reasons why I always encourage you to audio record the lectures, because it's impossible to pay attention for 4 hours while I'm up there yammering. It's impossible to pay attention for 4 hours when you're sitting at home, especially when you have all sorts of distractions and loved ones and all sorts of other things going on around you. It's very easy to be distracted. So having the opportunity to go back and view these things, go over it a second time, and more importantly, use all the other resources. I do my best to explain these things to you in the way that it makes sense to me. And hopefully that helps it make sense to you. But sometimes hearing these things in a different voice, whether it's reading it in the textbook, or it's watching that A&P Flicks video. Maybe watching that video with the great animations and the way she describes it helps this information to click. Studying this the most different ways you can is the best way to make sure you find the way that is going to work correctly for you. And that's really going to be the challenge over the next couple weeks, whether we end up back in the classroom or not. And we hear everything in between, right? Our president wants the world up and running again by Easter. We have other people at the CDC saying that it could be months. I have no idea. All I know is that our district doesn't have any idea either. They're planning for all contingencies. They're talking about what happens if we go back on campus after spring break. They're also talking about what happens if we have to do summer school online. What will that mean for lab classes and things along those lines? So nobody knows what's going to happen. All we can do is to do our best to learn this material, go one day at a time, and do the best we can. So this is going to be our challenge for the next couple weeks, but I know you guys are up to this. All right. So now you have a game plan. You know what you need to do to be successful. I will start doing office hours. So what that means is about 11:15, I will come on to the classroom a little bit early in here. So if there's any questions about the lab stuff or going over the things that we covered in the last class, you guys will have the opportunity to do that. And then my other class, I will have office hours for them as well. But also, if those don't work for you, or if you have something else going on, or you just have a question, email me. I'm very good at responding to emails. But if you need more than that, then like I said, I do have my own I have my own Zoom classroom where we can meet, and so we can discuss things, this way, or you can show me a picture of something that you have, or your video of something that you're working on, or whatever. I want to help you to be successful. So again, this is a new process for all of us. So I'm here to help in as many ways as I possibly can to help you make sure you get this information. All right, so any more questions, general questions for everyone before? Can we talk to you for a few minutes? Yeah, absolutely. So yeah, so if you have any questions, I'm happy to answer those and go over that stuff. If you have any questions about your exam or any other materials that you want to go over, I'm available. Like I said, I'll stick around for as long as people have questions, or like I said, I can meet outside of this as necessary as well. All right. All right, you guys, excellent job today. I know today was a rough day. We covered a lot of material, but hopefully it makes at least some semblance of sense, and then hopefully we'll moving forward. All right, take care. I'm going to stop the recording now, and I will see you guys in 2 days.